Hello students, am I audible and visible? Tell me quickly in the chat box. Hello, good evening, good evening. Share with your friends quickly and ask all of them to join. Let's see the chats moving. Yes, good evening. My voice is clear, audible, visible. Yes, I can see a lot of hearts. I can see a lot of hearts. Let me also check one, students. Hello. Yes, share it with your friends. Students are very less. Why? Being Saturday. That too being Saturday. So many less students. Hello. Yes. Yes. Hello students. Clear. Fine. Let's start our class. So welcome to Vedantu NEET English. I am Miss Gopika, your master teacher. So today we are going to do neural control and coordination. My God, very important chapter, students, mainly because digestion is removed and that reflex action, sense organ, eye, ear is removed. You are the luckiest batch because eye and ear is the most difficult concept. You know, if you're a dropper, if you have studied it last year, right, or you have been preparing for 2 years now, you all know that it is difficult, right? It is difficult. So that's the reason I'm telling you, you're the luckiest batch because that portion is removed from your syllabus. So shall we start the class? Are you all excited? I'm very excited. Yes, can I see lot of fire emoji in the chat box and can I see the likes equal to the live? Don't be conjuice, yaar. How many times to tell? How many times to tell? This dialogue has become famous but my students have not changed. It has become like one syndrome, right? Yes, okay. Students, so this is the plan. So before the syllabus reduction, we would teach this chapter in 3 parts. What are the 3 parts? We would teach neuron, okay? We would teach types of neurons. We would teach action potential, brain. We would also teach eye and the ear. But since it is removed, we are not going to learn, okay? We are not going to learn. Yes? They're telling, yes ma'am, it's difficult to like. Oh my God, I don't know what to even say. So are we ready to trick your brain? So this is the basic. You have studied in your 10th, you have studied in your 11th, maybe you have also studied in your 8th if you have done ICSE and all that. So let's just trick your brain. So I want answers from all of you and keep your score. Keep your score. Most of you got 12, some of you got 8 from the previous class, okay? So in the end of the week, I will ask you your scores. So that's why keep it ready, keep your scores intact in your brain, okay? So students, I want you to label this 1, 2 and 3. Quickly label 1, 2, 3 and take your 6 marks, okay? 6 marks question, 6 marks. So quickly answer 1, 2 and 3. Label your parts and let me know in the chat box what is 1, what is 2, what is 3. Okay, and we will start our session after that. Okay, can it be quick? Yes, go for it. Go for it. Very good, Rupa. Yes, 1, 2, 3. 1, 2, 3. Easy to label, right? Easy. We have learned this in our 8th, 9th, 10th, 11th. Now 12th again we are learning, right? 12th again we are learning. 11th also, neural control and coordination, 11th syllabus. Only Rupa has answered? My God, my students doesn't know. Very good. Yes, search and find. What a name, yeah. Search and find, wow. I should search you. Let's go, faster. Number should increase. Yes, correct. Very good, very good. Okay, I think my chats are running slow there, but here I can see a lot of them answered. Subhashni has answered, Rupa has answered, Dr. Strange, very good, Sanvi, Shivani, Nithya, very good, very good. So calculate your scores. Okay, calculate your scores. So first one is what? Your myelin sheath, right? Myelin sheath. This in detail you will learn. In detail you will learn this. Schwann cells. Okay, that secretes myelin sheath, you will learn today in this chapter. And then third one is your dendrites, right? Dendrites. And second one is your axon terminals. Okay, axon terminals. You can also call your synaptic bulb, synaptic knob. you can call any of this, or else simply you can tell Axon terminal. Okay, so good job on all those who answer. So quickly share the link with your friends and on your Telegram and on your WhatsApp and ask them to join quickly, so we can make the session more better. More the people, better the class. Okay, students, like last time, we are following the same pattern. The chapter weightage we are expecting this year for this chapter is around 4. That means 4 questions from this chapter, that is around 16 marks. Okay, why I'll tell you 4, because you can see other places it was 1, 2 and 3. This year we are expecting, because the syllabus has been reduced, they always pick on chapters which students actually find a little difficult, right? So we are expecting again 16 marks. Can you afford to miss the chapter? No, definitely not. Okay, full story done. You tricked your brain, got 6 marks. Very, very good. Very good, those who got 6 marks. Okay, students who are late, I am so sorry. You should have joined early, because your trick your brain question is over and we have already answered. So there's no point of going back. Okay, students, so today's chapter we are mainly focusing on our new nervous system, brain and the parts of it. Okay, so now already all of you know that our nervous system means everything to us, right? It is transported everywhere. It is there entire our body. Now you will be asking, ma'am, then we also have our endocrine system, right? We also have chemical coordination happening. That means hormones are doing the job of going everywhere also. But you tell me, which is the fastest system? That means, what am I asking? I'm asking if NS and your endocrine system, that means your chemical coordination, right? Which is faster? This system or this system? Which is faster? I would say, oh, Shivani, okay, new student. Hi, new student. So from next class, you have to join on time, because you will be having trick your brain question where you will get 6 marks, 4 marks from each question. Okay, so for that, students, all of you tell me, is endocrine or your nervous system going to be fast? Endocrine is going to be fast or nervous system is going to be fast? Okay, most of you have told nervous system. See, students, understand, when it comes to endocrine system, it is going to release hormones. That hormones are going through with the help of blood. It is transported all the places because of blood. So endocrine system actually reaches the target organs and it reaches every nook and corner of the body. Correct? But nervous system at the other end is going to transport everything with the help of electrical impulse. Right? Electrical impulse. That means electricity or this energy, ions, that is getting passed. That is telling that, oh, Gopika ma'am is teaching, so let's all join the live, right? That kind of impulse is sent by who? Nervous system. Okay, so most of the students have this doubt, why we have nervous system, then why should we have endocrine, or why should we have this chemical coordination? That's because hormones are easier to be carried with the blood to every place. Even though nervous system is faster, this will go faster. That means this will go to every nook and corner of the body. Correct? Because blood goes everywhere. Wherever blood goes, hormone also goes, right? Blood, I'm not able to say because it's become tongue twister now. Okay, let's understand this chapter. Okay, students, are you all with me? Yes, lot of you have joined. Very good, very good. Lot of you have joined. So, nervous system. When we talk about nervous system, students, what do we remember? Nervous system. When I talk about nervous system, I remember about only one person. And the one person that I remember is neuron. Whom do I remember? Neuron. So neuron is what? Neuron is the basic structural and functional unit of a nervous system. So shall I write that? Right? It is the basic structural and functional unit. Correct? Unit of nervous system. All of you agree? Right? We study about cell, right? We studied about nephron. So all these are the basic structural and functional unit of whichever system we are studying. That means without them, nothing can happen. Correct? Without them, nothing can happen. So is this clear? Very good. All of you have answered. Very good. Okay. This is the structural and functional unit. Now, when we talk about neuron, or when we, tell about neuron, what do we learn about neuron? Students, about the nervous system. See, your NCERT goes in an order where they explain the coordination, then they explain the nervous system, and then they explain the neuron. So, I will be going in an order where I explain neuron, then when I teach you brain, right? Starting of the brain, I will teach you your nervous system. Okay, that's the order we go. Okay, yes, very good. Now, when we talk about our neuron, can I divide neuron into two main parts? Yes, ma'am, you can divide neuron into two parts. You can divide into how many ever parts you want, because only students who are ready to learn will be bothered about how many parts I divide. Those who have come here for spam, they will not bother, correct? Are you all with me on this? So students, I am splitting neuron into two parts. What am I splitting neuron into two parts? I am splitting neuron into a cell body part, okay, cell body. I want all of you to tell me the other names for cell body. We have many names for cell body. I will write two names, other two names you people will tell, okay, other two names you people will tell. Come on, cell body, we can also call it perikaryon, okay, other two cell body names you people will tell, okay. And cell body I divided, it can also be called as perikaryon. Here I am dividing this into a long unbranched short branch structure, okay. I am dividing the neuron again into a long unbranched, okay, long unbranched, and here I am dividing it into short branched. Very good, amazing, okay. It is also called as cyton, right? We have all learned this name. It's also called cyton. It's also called soma, right? It is also called cyton. It is also called soma. Today there was a question on the Telegram channel. I don't know how many of you answered it. The question was asked about protein synthesis, like where does this protein synthesis happen, or which is the part that is capable of forming proteins. I think very few of them answered. The answer was soma, that is your cyton or your cell body. Today's class you will understand why, okay. So if you are not a part of our Telegram, please join our Telegram, okay. Yes, students, here I have split it into two parts. What have I split it into? Long unbranched, short branched, okay. Long unbranched is what? All of you know this. It is long and it is unbranched. What is it? Axon, right? Axon, okay, axon. And short but branched, dendrite. All of you know this, basics, dendrites, okay. So this is how I am going to split, or this is how I am going to separate your neurons, right? Yes or no? Students, no spamming, come on. You can ask how each of you are later after the class, okay, after the class, because if you keep spamming I will not be able to see if you answered, right? Long unbranched axon, short branched dendrites, right? These are the receiving. Dendrites are the part which receives the impulse. Gopika is going to touch a hot object. How is the impulse coming? Impulse is coming from the dendrite, moving on, right? Moving on to the axon, from the axon to the next dendrite. It keeps moving, right? And until I know that, okay, I have to retrieve my hand, right? I have to retrieve my hand. So this is the basic. This is the basic. 1 second, students. Yeah. Okay, this is the basic of your, what did I tell? Just the general. Now, if I have to understand the neuron, first we will draw the neuron. All of you draw with me and then we will understand it, okay. All of you should draw with me so it is easier for you also to understand, and when I label you will not be looking around, oh what is ma'am drawing, what is ma'am labeling. Fast, you can draw with me. So yes, students, I'll show you once I finish drawing. Yes, all of you draw with me. So at the end of the class we both will have beautiful set of notes, and later you can download the notes also, okay. Yes. Done? Done. Neuron looks good. Neuron looks good. Zen, don't shoot into the gun. I haven't even reached spinal cord. I haven't even told a word called involuntary action. So let's go step by step. Right? Yes, students, all of you drew the nervous neural. Yes, thank you so much. Okay, so students, this is what we have drawn. Okay, let's label the parts which we already know, and then let's later label the parts which we don't know. Okay, let's later label the parts which we don't know. So I am putting some dots on the cytoplasm, because cytoplasm has all the organelles like your mitochondria, SCR, ER and all that, right? Yes. Okay, yes. So students, let's label the parts we know, we just now discussed. So this becomes what? This portion becomes your cell body, right? This becomes your cell body or your cyton, right? This long unbranched structure, what does it become? It becomes your axon, right? Axon. And this portion becomes your axon terminal. I don't think any student has a doubt with this. Axon terminal. Okay. Oh no, how will I label, students? Let me draw it on this other side. Okay. Wait, I'll draw this on the other side. Yes. So here, can you all see if I draw it here? Yes, you can all see, right? Students, cyton. Okay. Cell body. Students, always have 2 color pens when you are attending any kind of biology class, because it's easier for you to differentiate. That's why I always write with many colors, just so that my notes look good for you people and it's easier for you to learn. Okay, axon terminal. Labeling done, that side. Let's start labeling this side. Okay, yes, this is what? Simple dendrites. Okay, this is what? Nucleus. This is axon, already labeled. This portion, cytoplasm. Cytoplasm has what and all? It has all the whatever organelles are required, but it will have mitochondria, SCR, RER. Okay, so this is, I'm only writing here SCR and RER. I'll tell you why the importance is. Yes. Okay. Now this is general. Nothing else I have drawn extra. Nothing else I have drawn extra. Okay. Now students, we all know that there is some sheath-like structure, right? There is some cells. There are some cells on this axon. Axons are not always like this, right? What are those cells called? Schwann cells. So let's draw that cells also. Let's draw that cells also. Right? All of you have seen the cells without even knowing. When we were in school, we used to draw the cells, right? See, like this. We have drawn the cells. Okay, we draw. Yes. Now I have drawn Schwann cells. Now this Schwann cells, okay, let's label the Schwann cells also. This Schwann cells will secrete something. Schwann cells. Students, this Schwann cells will secrete something white color, okay, a white color structure inside, okay, like this. It will form a white color structure inside. It will secrete a white color structure inside, which is called your myelin sheath, which forms like a sheath-like matter. Did you all know this? Did you all know this? You can all see? Okay, fine. Okay, this white matter is called as your myelin sheath. Did you all know this? Schwann cells give out or secrete a white color sheath-like matter, which is called the myelin sheath, which actually protects, which acts like a shock absorber, right, for the neuron. Yes? Okay, fine, great. Now students, after this, there is one more very, very important. I'll draw with this color. Here, okay, here, there is going to be something called as your axon hillock. Okay, axon hillock, which is the most, okay, axon hillock. Now axon hillock is the most sensitive part of the axon. Why am I trying to say this? That means an impulse is coming. Okay, imagine Shalini. Okay, Shalini touched a hot object. Okay, she was in the class, she was listening to my class. She touched the hot coffee which was kept on the table. the moment she touched it, or she was about to touch it, she could feel, right? Our sense organs could feel the heat. That impulse has been sent, right? The next impulse that has to come from the brain is retrieve your hand. For that, see, when any impulse is coming, this axon hillock, the most sensitive part, will collect the impulse and immediately transfer. Okay, that fast transfer, so that this person does not get burnt, right? Shalini will be like, oh, this is very nice, so warm, let all my fingers get burned. After that, let my brain send signal, then I will remove my hand. Can anyone wait for that? Can anyone wait for that? No, right? If a teacher is walking in the class, you are copying, immediately teacher does this, by then you will move. How is that fast? How is the impulse fast? All because of your nervous system. Okay, so axon hillock is the most sensitive part on the axon. Yes, clear? Okay, then here, students, here, this portion where you're inside your axon, okay, the cytoplasm of your axon is called your axoplasma. Okay, and then this outer layer, right, outer layer of your axon is called what? Can anyone tell me? What is the outer layer? Axolemma, right? Axolemma. So axoplasma, and we have axolemma. This is the outer layer, axolemma. Clear? If I write like this, that means the outer layer of the axon. This white color which I have drawn, the outer layer of the axon. Okay, yes, great. Okay, then students, one special people are there. So I told you in the cytoplasm, RER is there, right? Rough endoplasmic reticulum. Now, can anyone tell me the function of rough endoplasmic reticulum from your cell? Can you remember? Cell, what was happening? What is this rough endoplasmic reticulum going to do? By then, I will draw something. Okay, I'll draw something. See, I'm drawing your some kind of granules. Okay, the blue color dots which I have drawn. Very good, Rupa, very good. Protein synthesis, your rough endoplasmic, Sanvi, good. Rough endoplasmic reticulum can form proteins. So, that rough endoplasmic reticulum which I have drawn here, blue color, those are called as your Nissels granule. What are they called as? Nissels granules. So, what are they capable of? They are capable of forming protein or synthesizing proteins, and these proteins will help in the formation of one category of neurotransmitter. It will help in formation of one category of your neurotransmitter. Is this clear to all of you? Nissels granules, or your rough endoplasmic reticulum, which is only present in the cell body and it is moving towards the dendrites. If you see, it is going towards the branches, are capable of forming protein. That protein, you know, neurotransmitters are a type of protein, that one category of neurotransmitters will be formed by your Nissels granule. Okay, Nissels granule. Now, can any of you tell me the difference between axoplasm and cytoplasm? Can you tell me the difference between axoplasm and cytoplasm of an neuron? Come on, tell me someone who can tell me this. Axoplasm and cytoplasm of the answer. Just look at the thing, you will get the difference. Just look at it, you will get the difference. Come on. Yes, is there anyone? Very good, Sai, very good. Okay, presence of Nissels granule, right? Presence of Nissels granule is only seen in the cytoplasm of the neuron and not in the axon, So who can do protein synthesis? It's only the cell body, or it's only the cyton. So students, I told you proteins are formed which will form one category of neurotransmitter. That one category of neurotransmitter will be transferred where? From here, it will be transported to your synaptic knob. All of you remember this, you have all studied this. It will be transferred to your synaptic vesicle or your synaptic knob. Okay, synaptic knob. From your synaptic knob, it will be transferred to your synaptic vesicle. Synaptic vesicle. So your neurotransmitters will be stored in the synaptic vesicle. So if I draw an enlarged structure of this, how will it look like? How will it look like? It will look like this. This is your synaptic knob, right? Synaptic knob. Inside that we will have your synaptic vesicle. Okay, inside that you will have your synaptic vesicle. Inside that you will have your neurotransmitters. Clear to all of you? Is this clear? Crystal clear? Okay. One 3D I took and drew. Okay, one 3D when I drew, this is your synaptic knob. Inside this you have synaptic vesicle. Inside that neurotransmitters will be stored. Okay, any doubts on this, students? No doubts, right? So this Nissel's granule, we know that this is only present. So I am writing here, okay, Nissel's granule can synthesize protein and it is only present in the cell body plus the dendrite. Cell body plus the dendrite. Yes, very good. All of you drew? Did all of you draw? Or is there anyone who didn't draw? Who's waiting for my notes to come? No ma'am, you draw, I will draw later. Is there anyone like that? No, right? Yes. Very good. Neurochemicals, right? Yes. So students, this is clear. Now, is there necessity that all the time myelin sheath should be there? There can also be axon where the myelin sheath is not there, right? That means what? I can also have, right? I'm writing it in a box here. That means it's not necessary that myelin should be there. If myelin is present, that means myelin sheath is present or secreted by your Schwann cells, right? Then I will call that as myelinated neuron. This also all of you know, myelinated neuron. Okay. If myelin sheath is absent, non-myelinated, simple. No myelinated sheath or myelin sheath. Simple. It's called non-myelinated axon. Non-myelinated neuron. Okay. Myelinated neuron. Are you all with me on this? Yes. Clear. Okay. Now students, understand one more thing here. One more thing that you need to remember in this. Okay. This is all clear. This is all clear. We also need to understand. Okay. We also need to understand, students, in this, okay, in this full thing, your CNS and PNS is there. That means central nervous system is there, peripheral nervous system is there. In central nervous system, okay, again I'll write it in one more box. So you people will not be like, oh ma'am wrote everything together, I can't understand. Okay, here, kutti box, this much, yes. Okay, students, look here. In your, what I was telling, CNS. In CNS, do anyone know who makes the myelin? Who makes the myelin? Yes, very good. Okay. Who makes the myelin sheath or myelinating the cells? Who will be doing? Starts with O. Oligodendrocyte. Anyone knows this? Oligodendrocyte. Oligodendrocyte. In PNS, your Schwann cells makes the myelin. Clear? Clear? In CNS, okay, in CNS, oligodendrocyte will form the, yes, oligodendrocytes will form the myelin sheath. That means they will go through proliferation, differentiation, myelination to finally form your myelin sheath. But at the same time, in your PNS, that means your peripheral nervous system, your Schwann cells will give out the myelin sheath. This is all about your neuron. Okay, I haven't missed one point. I have also given you some extra points. Okay, I have also given you some extra points. Is there anyone who has any doubts? I hope there is no doubt. Okay, yes. So students, when we spoke about neuron, we were telling about what to tell, the structure and everything. But what is the basic function of neuron? What is the basic function of neuron? Simple, to transmit the impulse from one part to the other. Correct? So if I draw a neuron, okay, this is a simple neuron. Okay, I am not drawing a proper structure. Okay, this is a simple neuron. Okay, this is one neuron. Students, now what is going to happen? One impulse is going to come, right? One impulse is going to come and it will start moving. It will keep moving, go down, right? Go down to the axon terminal. From here, another neuron, right? Another neuron, so we will draw it in another color. Another neuron's dendrite will be waiting to receive it. All of you know this much? Head of one neuron and tail of another neuron. Head of one neuron and tail of another neuron is connected, right? All of you know this also? Okay, so this is how it happens. So what am I trying to say is, if head of this neuron is connected to the tail of this, right? Tail of this. So tail of one neuron, head of another neuron, tail of one neuron, head of another neuron. So they are actually connected like this, but it will not be like this. It won't be in linear structure. It will be like this. Okay, head here, tail here, connected like this. It will go. Okay, like this it will go. That's why if you search for neuron transmission or nerve impulse, you will see structures like this. Okay, yes. So how is it going? If I have to write the pathway, how is the pathway going? All of you tell me. How is the pathway? How is the pathway of information going from the neuron? It is starting from, let's write, it is starting from dendrite, right? It's starting from dendrite. Dendrite, it is going to the cell body or your cyton. From cell body, it is going to your axon. From axon, it is going to your axon terminal. Any doubt in this? Right? This is how it's going, correct? This is the pathway. So the impulse is coming. It is going from the dendrite, cell body, axon, axon terminal. Done, 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 done. Okay. So let's write the function also. So what is the basic function of a neuron? Simple. To transmit impulses, right? To transmit impulses from one part to another, one part to another. Simple. That's all. That is the function of this person. Function of whom? Function of our neuron. Okay. Any doubt so far? Let me know. Students, now types of neuron is there, right? Types of neuron is there. That means, see, if I draw this neuron, say I told you impulse is coming from one place, it is going through the dendrite, it's going to the cell body, it's going to the axon, simple, okay. But there are chances that, see, from here also the impulse is coming, from here also impulse is coming. So imagine now Subhashini is drinking a coffee. So she is seeing the, what do you call for that, students? The smoke, right? The fumes, I kind of forgot the word, okay, the smokes are coming out of the cup, right, cup of your coffee. So she's visually seeing it. She's like, okay, this is hot, okay, this is really hot. By touch also she is able to sense it, that okay, this is hot. Now two impulses are coming. That means from your optic nerve, that's from your eyes also, from your sensory also, two connections are coming, that oh, it is hot, right? When like that multi impulses come, maybe when we are walking, we are also talking, when we are so many multitasking we do, right? That time, that kind of a neuron is called as multipolar neuron, and it is present in the brain. Okay, multipolar neuron. Okay, multipolar neuron, that means it's present in the brain. What are they actually doing? Students, I'll write the where it is present here, and I'll show you the diagram in the next slide. Okay, where is it there? It's present in the brain. That means from many places impulses are coming, from many places signals are coming, and it is getting transferred. That is called as multipolar. Vapor is, yeah, vapor. Thank you, Lakshmita. Is it called vapor? Yeah, vapor, I guess. Students, that, I don't know what you call that, yeah. I can't still remember. I can't still think of it. Okay, multipolar. That means many signals are coming. Now, unipolar can be there. Easy, you know. If multi is there, uni also is there. That means what? From one way, only one direction signal is coming. That means only one axon is there. From one cell body, only one axon is there. One signal is coming, right? That can be called as unipolar. I'll show you the diagram. Do not worry. Just write it as of now. Okay, unipolar is there. Now where is unipolar seen? It is mainly seen in lower invertebrates. We cannot afford to have it, right? So it is seen in lower, if you write invertebrates only, it is lower, or you can write lower organisms. Are you all with me? Are you all with me? Any doubts so far? Yes, yes. Hi, Vijayan. Hi. Yes, I do remember you. I always remember my students, okay? It's not like I forget my students, okay? I always remember my students. Students, any doubts so far? Any doubts? No, right? If no doubts, put fire emoji in the chat box. Otherwise, we are good to go. Yes, very good. Multipolar neuron is in the brain. Unipolar is seen in lower organisms or in invertebrates, okay? Now, I told you that. Now, students, since multi is there, uni is there, bi also will be there, bipolar also will be there. Now, what is bipolar? I'll show you the diagram that, in the next page, but in case if you want to have, if you have a doubt, see, this is how bipolar will look. That means from one cell body, one axon, one dendrite. One axon, one dendrite is going. This is called as bipolar. Bipolar. That means from one cell body, one axon, one dendrite. One axon, one dendrite is going. Clear? Very good. Yes. Chats are filled. Very good. Yes. So students, bipolar. Clear bipolar? Okay. Yes. Bipolar. That means from one cell body, one axon, one dendrite is going. So bipolar is present in the retina. Okay. It's present in the retina or in your eye. Right. Retina is there where? In the eye. Yes. Okay. Last one is your pseudo unipolar. Students, what is the meaning of pseudo? I have told this in animal kingdom. Pseudo means what? Pseudo. Very good. Retina of the eye. Thank you, Vijayan. Students, retina. Pseudo means what? Tell me. Pseudo means fake, false. Correct, fake, false. So pseudo unipolar means this one, no? So it looks, okay, let me draw it like this only. Students, it was going like this, one cell body, one axon was happening. They thought, okay, this is going to be unipolar, right? This is going to be unipolar, but suddenly it made extensions. Right? Your brain will be like, yes, today you are going to sit for the full live. Even if it's 3 hours, even if it's 4 hours, I'm going to sit with Gopika ma'am's live. Suddenly your brain will be like, no, I'll watch Instagram. I'll watch one Netflix series. What is going to happen? What is the big deal? Neural control and coordination will be there forever on YouTube. I will see before the exam. Right? I have still so much time. That is called as pseudo promises. You make pseudo resolutions in your brain. Right? That is called pseudo. So here students, what are they doing? They made off one pair, right? They made off one extension suddenly. So that is why they are called as pseudo unipolar. Where are they present? In the ganglions, right? In the ganglions of what? Of your spinal cord. Okay, spinal cord. Dorsal ganglion of your spinal cord. Spinal cord. Dorsal. Okay, I'm writing here. Dorsal. Yes. So we have understood types of neuron. What is neuron? How is neurotransmitters formed? Then pathway. How is it connected? Everything is done. Done? Are you all with me? Done? Okay. So students, look here. This is what I was talking about. Okay. So this image is clear? Yes, very clear. So bipolar, can you see? There is one. Okay, wait, I'll use red color. Okay, students, so can you see? One cell body is there, right? One cell body is there. From one cell body, one axon is coming out. One dendrite is coming out. Right? Dendrite is coming out. Yes? Clear? Oh, I don't think this color also is clear. Students, I'll write it with black only then. Okay. Yes. So look here. One cell body, right? This is the cell body. From then, one axon is coming, one dendrite is coming. I've added this image only for your understanding, because in your textbook it is only mentioned multipolar bracket retina of the eye. So it is mentioned very lightly. So students, look here, what is this? Pseudo. So that means, see, from here we thought, oh, this is going to be unipolar. Suddenly it made of branches. It suddenly made one axon and it suddenly made one dendrite. So this becomes pseudo. Look here. This is a normal one, right? This is the normal one which we drew. Multipolar. From many places impulse can come, and from many places they can also receive it and pass on to one, right? This is called as multipolar. Now look here. Pyramidal cells. This is not there in your syllabus. Okay? Pyramidal is not there in your syllabus. Yes or no? Yes. Fine. So this is multi segments it's going. See, many branches. Again, many branches. Even in the cell body there are branches. Okay. So this not part of your syllabus. Yes. Students, now I told you everything about neuron. If any student has any doubt about neuron, this is the right time to put it in the chat box. I will see your messages. Okay. Now students, this is one neuron. This is another neuron. This is another neuron. Okay. Let's also, no one would have ever seen a neuron like this. Okay. This is jugaad, right? Jugaad neuron. This is, otherwise it will take a lot of time if I draw like this 4 neurons and come. So that's why we will understanding concept is important, students. Diagram. Okay. So this is your new type of neuron which I have invented. So yes. So look here, all of you. Simple. One impulse is coming. Okay. One impulse is coming from here, and that impulse is getting carried away. Okay. Impulse is getting carried away. Now what did I notice? Okay. What I noticed is, one second. Okay, students, this is your axon. This is your axon. Okay, students, what I am noticing here is that there is a gap. There is a gap between both of my neurons. Okay, both of my neurons, there is gap. So can you see here there is one gap, here there is one gap. Okay, so if an electrical signal has to keep passing on, but there is a gap, my signal will get blocked, or my signal will not be completely transmitted. So what I am trying to say is, throughout this area, right, throughout this area, there is going to be an electrical transmission. Okay, there is going to be an electrical transmission. Now what am I going to do about the gaps? What am I going to do about the gaps? In the gaps, chemical synapse will happen, or your chemical transmission will happen. Is there any student who has a doubt? Easy to understand. Chemical transmission will happen. Okay, so electrical transmission is happening. That means one neuron is there, one neuron is there, one neuron is there, but there are gaps in between the neuron. What to do while there are gaps? In that gaps, chemical transmission will happen. Okay, chemical transmission will happen. Okay, yes, chemical transmission. Yes. Again here what is going to happen? Oh, 2 pencil writing. Students, here also what is going to happen? Electrical, chemical, electrical, chemical. Clear? Simple? Easy? Okay, easy? Yes, very good. Yeah. So once this is clear, let's try to understand one more thing. Now here, wherever the neuron was there, okay, wherever the neuron was there, topic is deleted, Vikram, sense organ topic is deleted. How many times have we done? No more syllabus confusion. Go check that video out. We have clearly told sense organs, reflex action, eye and ear is completely removed. Okay, your lucky batch, please don't manifest that NTA brings that back to the syllabus. Okay, it's going to be difficult. Okay, yes students. So students, when I say electrical transmission is happening, chemical transmission is happening, can I call this both together as electrochemical transmission? Electrochemical transmission. Yes or no? Electrochemical transmission. Easy? Easy? Exactly. I'm going to stick it on my head. Students, this is the deleted syllabus. On my hand, this is the deleted syllabus. This is the, I'll print it on the t-shirt also. This is deleted, this is deleted, this is deleted at this point. Because people ask me every single class, how many of our videos we do is not enough. You won't watch the videos, later you will come and ask in the class. No? Yes or no? Yes, electrochemical transmission. So wherever gaps are there. Now I told you gaps are there, but I didn't tell you how much gap is there, right? So students, if the gap, so here earlier, right? If the gap is more than, okay, is more than 2 nanometers, it will become chemical synapse. It will become chemical synapse. Easy? Easy? So this electrical transmission, right, this electrical transmission is happening because of your ion exchange, right? That is where you're going to study action potential, right? That is where you're going to study action potential. That is where you're going to study what? That ion exchange. How is this electrically happening? How is this moving? All that you're going to study here. So here is where you're going to study your action potential. Okay, I'm just giving you an idea. I will teach you what action potential is. Okay. This is where action potential will happen. Now, wherever this chemical transmission is happening, here is where you will talk about synapse, right? Synaptic cleft, synapse, all that you will talk about. Your neurotransmitters, right? So, I'll write neurotransmitters also. Neurotransmitter. So, is this clear? This is how your chapter is divided. This is how your chapter is divided into chemical transmission, where you will study your action potential, and here in the chemical you will be studying about your synapse, neurotransmitter, how these neurotransmitters are transferred. Okay? Yes. Okay, let's try to understand this. Image is clear? Yes, image is clear. Okay. Yes, yes. With me? Tired? No, no. Easy. Easy topics we did so far. Now here, this topic you need to give your full attention, students. Even for 1 minute if you blink, no, you will not understand, because this topic you have to continuously try to understand. Oh, this is getting opened, okay, this gate is going there, ions are moving. This concept is very, very important, and it is, I don't think, see, it's not easy for everyone to explain this concept, okay, and it is not easy for everyone to make you understand. That kind of a concept this is. Are you all with me on this? Okay. So students, look here, I will first explain you this with the help of a diagram. Then I will explain you with the help of my own diagrams. Okay, simple. Let's make it very simple. I told you electrical transmission happen. That means from one neuron to another neuron, electrical transmission will happen. Okay. Now this electrical transmission will happen if there is no gap at all, right? No gap is there, and between one neuron to another neuron, electrical transmission will keep happening. Okay, so this much is fine. Now in the resting phase, that means no impulse has come. No impulse has come. Full resting phase, the neurons are there. What is actually going to happen? Okay, look here. The resting phase. Here we are talking about your. See, all of you, look here. Look here, students. Here I introduced you to axolemma and axoplasma. We are talking about axolemma. Okay, all of you have to be with me. We are talking about axolemma in the conversation of action potential. Okay, so simple. Just understand, in the resting phase. Okay, in the resting phase, outside the axolemma, positive charge will be there. Inside the axolemma, negative charge will be there. Simple, easy. Positive charge is because of sodium ions. Negative charge is because of negatively charged proteins in the inside. This much clear? This much clear? Okay. Potassium ions are there, but these potassium ions are not enough to show the electropositivity here. But the negatively charged protein ions are giving the negative. So this is called resting potential because this is polarized. That means outside positive, inside negative. Polarized, equal. Okay, you are angry, your sibling also is angry. Polarized. No fight is, no, nothing is going to happen. Even if fight is going to happen, both of them will kill each other or both of them will not talk to each other. Okay, if one person is pavam, one person is very calm, other person is very angry, some kind of energy exchange will happen, right? So same way here, outside positive, inside negative. How is the negative? Because of negatively charged proteins. Okay, here, fine. Now suddenly one impulse has come that YS or Niharika is going to okay, YS and Niharika are both of them are touching a hot object. Okay, impulse has come. When the impulse came, okay, when the impulse came, so resting potential, what happened? Outside positive, inside negative. Outside positive because of sodium ions, inside negative because of negatively charged your proteins, and your potassium also is there. Potassium is not enough. Okay, potassium is not enough. Now students, Niharika and YS is going to get into a fight, or they are going to grab the same thing. Okay, impulse has come. Suddenly what happens when the impulse comes? Your sodium channels will open. Sodium channels will open and they will start entering inside. Can you see? This is the membrane. Potassium, sodium channels started entering inside. When sodium channels enter inside, what is going to happen? This electropositivity is going to come inside. Can you see here? Positive, positive, positive. That means from negative it became positive. Why? Because sodium came inside. Simple chemistry. Simple chemistry. Higher region to a lower region, something has come. So charges also changed. Charges also changed. Here it became what suddenly? It started becoming negative because sodium reduced. Sodium reduced, right? I'll tell you about the voltage. Okay, I'll tell you about the resting potential. I'll tell you about the voltage when I am explaining it. Now just understand the diagram. Okay, students, then again, okay, here depolarization happened. Okay, depolarization happened. Then again repolarization will happen. That means they will be trying hard to come back, right? They have to go back to resting potential. For that time to happen, at one point when they reached maybe say plus 45 MV, when they reach plus 45 MV, sodium channels closed. Suddenly sodium channels got closed. Potassium channels opened. Potassium channels opened. Now what is going to happen? Potassium channels opened and they started moving to sodium. Students, simple, I'll tell you, simple. You and your sibling both are in resting phase. No one is harmed. Okay. Suddenly, okay, suddenly your sister got very angry that you took her dress or something, or your brother got very angry because you took his shoes. He'll start attacking you. He'll start attacking you because you are not attacking. You become the positive person. Because your brother is attacking, on his side, negative will come. Okay. Then he hit you so much that you lost your temper. He hit you so much that you lost your temper. Now who is going to attack? Your brother got Shanti Samatana. Your sister got Shanti Samatana, telling that okay, I anyway hit her. I am happy. Correctly, when she thinks that she is happy, will you let go? No, you will start going back to hit her back for whatever she gave. Correct? So now potassium channels open, they will start moving there. They will start moving there. So who becomes the negative person? Here. You become the negative person. That person becomes positive because they finished fighting. Aram se they went back. Okay? Then both of you, parents will come, auntie, uncle will come, everyone will come. Your aunt, your uncle, your next... what to tell, your neighbors will come listening to this, and finally both of you will relax and you will be like, let's go to resting potential. Let's go to resting potential. This is all your action potential. Students, I'll tell you, this is one concept that my teachers found it very difficult to explain it, maybe because we couldn't use such examples, right? So as a teacher, I wanted to make you explain it this way. I hope this is clear to all of you. Yes, clear? Okay, is this clear? Simple, easy. Now it's easy for you to understand the positive and the negative. Okay, so Na becomes your sibling, K becomes you. Okay, again here you are going to the sister's territory, right? So you become negative, sister becomes positive. Then finally family, everyone will come, and then you will be like, okay, let me go to the resting potential, where both of them will start going back to their places. Yes? Great, clear. Okay, now we will understand from our point of view, right? From our NEET point of view. Now you understood the concept. Concept is clear. Now we are going to understand what are the things that are present, how it is actually happening. Okay, how it is actually happening. So you should not forget to comment that there is, you put the time and show the students that see Gopika ma'am has explained it this way, right? So you people should comment. Every time I tell you, people comment like, no ma'am, not possible ma'am. Look at the likes, students. Why are the likes not increasing? You people are planning to bunk, huh? If you bunk, no? Students, likes should increase. Let it become 100 fast. Make it 100, make it 100. Let the likes. Yes, very good. Okay. Thank you so much. Thank you so much. Okay. Students, now I told you the story behind it. Now let's try to understand what and all is actually there. Okay. So I told you axolemma is there. Okay. This is your axolemma. So I told the outside, right? Students, you should think, when I tell sodium, you think of yourself. When I tell potassium, you think of your sibling. No. Potassium, you think of your, sodium, you think of your sibling. Potassium, think of yourself and try to connect. So students, look here. Here, what is happening? Positive, 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 positive. That means here, who is there? Potassium is there. Okay. This is axolemma. Okay. That also I'll label. Axolemma. Okay. Down, what is there? Down, what is there? Negative is there. Simple. Negative is there. Why negative is there? Because of the presence of potassium. Actually, potassium does not play a role, but negatively charged proteins. Negatively charged protein. Now, this is resting phase. This is resting phase. Right? Yes, you should make it viral. Let me see Dr. Strange. We'll see if you people make it viral or not. Okay. So students, sodium is present. This sodium is present in the extracellular fluid. ECF, extracellular fluid. Okay. Now, this is the resting potential. Now, I am writing here, this is the resting phase, right? Now this resting potential is going to be minus 70 MV. Okay, minus 70 MV. When it is minus 70 MV, sodium is outside, potassium is inside. Both are sleeping peacefully, no disturbance at all, right? No disturbance at all. Yes, more proteins compared to the potassium. That's why potassium sign is not shown. Here electropositivity is sodium and negativity is because of the protein. Resting potential minus 70 MV. Okay. Here no doubts are there. Now, I told you, when we regulate this, right, when some changes are happening, okay, this axolemma, okay students, okay, I've drawn it like this. They have some leaky channels. Okay. Okay, look here all of you. This is your axolemma. They have some leaky channels. Leaky channels means some or the other things will keep passing in and out. Maybe potassium goes out. Okay, maybe potassium goes out. Sodium comes in. Okay, this is leaky channels. This leaky channel, something or the other always keeps leaking. Okay, but it does not make a very major difference. Okay, major difference can be because of a sodium and a potassium pump. This will help in pumping. But for sodium and potassium pump to get activated, or for sodium and potassium to pump, they need one fellow. Who is that? This fellow needs, one second. This fellow needs ATP. It needs what? ATP. So when one ATP comes and this is planning to pump, okay, this is your NCERT line, very, very important, NCERT line. When one ATP comes, it can give out 3Na and it can take in 2 potassium. What did I say? When one ATP comes, okay, when one ATP comes, it can take, okay, one ATP comes, it can give out 3Na plus, and it can take in 2 potassium. Are you clear on this? Are you clear on this? One ATP, how many sodium and how many potassium can it move in and out? Question can come. Okay, question can come. 3 Na will go out, 2 potassium will come in. Okay, 3 Na, look at the arrow marks carefully. 3 Na will go out, 2 potassium can come in with the help of 1 ATP. So every time the pump has to get on, ATP has to keep coming in. That is why you also need energy. Otherwise your impulse also will not get transmitted. Okay, fine, very good. Now these are leaky channels. Okay, these two are leaky channels. Leaky channels, something or the other will always keep leaking, and this is potassium and sodium pump. Okay, now resting potential clear? Resting potential clear? Let's try to draw what will happen if an impulse happens. Okay, so I'm drawing the same thing here. Okay, positive, 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 positive. Okay, that's your sodium. Okay, sodium. Yes, students, here an impulse came or a stimulus. Stimulus. Okay, one stimulus came. This is your resting potential. Suddenly one stimulus came, and that stimulus voltage is around minus 55 MV. That means the stimulus has to be in the threshold. Stimulus has to be in the threshold, and that is minus 55 MV. So when a stimulus came, what did I tell you? What did I tell you? What is the stimulus? You wore your sister's dress or your brother's shoes. That is the stimulus. Stimulus has reached the threshold. They have been controlling for so long. Now they reach the threshold. Now when they reach the threshold, what are they going to do? They are going to open the sodium potassium pump. That means the sodium gated channels. Okay. So I'll draw that. I'll draw that. Okay. Okay, students, this is how the channel will look. This is your gate. Okay, this is your gate. This is your sodium gate. So it was closed. It was closed during the resting potential. Now suddenly one stimulus has come. That means what? One trigger has come, right? So suddenly this gate will open. When the gate opens, what will happen? Sodium will start coming in, right? Sodium will start coming in. Okay, sodium will keep coming in. Many sodium will come. Okay, now what is happening? Sodium is moving from a region of higher concentration to a region of lower concentration. Now what is going to happen to this action potential? What is going to happen to this action potential? Let's draw negative. Negative. Here what is happening? Positive. Wherever the channel opened, right, there exchange of signs are going to happen. Is this clear? Is this clear? Yes or no? Yeah, lawn surprise if you people ask me now only, how will I tell? Right? Yes, very good. The gate opened because of the stimulus. So now the gate opened. So now what is going to happen? Here can you see change of signs have happened. That means here sodium has come in. Because the sodium came, here it became electropositive. Remember all of you? That means you were not fighting. You were not fighting. So near your side it became electropositive. Because your brother started the fight, he became electronegative. Clear? Okay, great. Great. Amazing. Okay. This much is clear. Okay. And this, wherever this exchange is happening, right? Exchange is happening. This is called as depolarization. This is called as depolarization. Okay. This was polarization. This is called as polarized. Okay, this is called polarized because both the signs are there, up and down. This is called polarized. This is called depolarization. Okay, this is depolarization. Now repolarization will happen. Okay, now repolarization will happen. So I told you these pumps are dependent on voltage. They will only open when the voltage will reach the threshold or some change is happening. If it is minus 70, these are closed. But suddenly voltage change happens, this people will open. Okay, now imagine here also some voltage change happened. Maybe it reached around plus 45. Okay, it can go up to, it can go from plus 45 to 65. Okay, plus 45 MV to 65 MV. Okay, during this time what is going to happen, students? Again, you and your siblings. Okay, so this is the sign. Okay, look what has happened. Plus, plus, minus, plus, plus, plus. Okay, now here plus, plus, minus, plus, plus, plus. Students, actually this plus and this minus, they both will try to get attracted to each other. This two will get attracted to each other. Yes or no? Negative and positive will get attracted to each other. So they will start shifting. They will start shifting. So what is going to happen here? What is going to happen here? From the third position, your negative will shift to the fourth position. Positive will come back here. Okay. So like this shift will going to continuously happen. Done? Done? This much clear? So here action potential is happening, right? One action potential is happening. Okay. Yes. Now that has happened, so the signs have been moving. Now repolarization has to happen. During repolarization, what is happening? Our potassium channels are opening. That means you got so frustrated, you got very angry that your brother came to hit. Now you are going to hit them back, right? So that time, what are the signs that is going to come? So here we have again plus sign, right? Plus, plus, minus, plus. This is shifted. Okay, this is okay. I'll draw that also. Okay. Minus, minus, plus, minus, plus, minus, minus. Okay. I told you they are getting attracted, so they are getting shifted. Okay, positions are changing. Okay, shifting is happening. Shifting is happening. They have reached this situation. Now repolarization has to happen. So in repolarization, your potassium gated channels are going to open. Okay, repolarization. Your potassium pump is going to open. Now potassium pump will open. Okay, so here again you will have positive, 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 negative, 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 negative. But there is a catch. The catch is that because potassium pump opened, here it's going to be potassium. Here is going to be potassium. That means what has happened, students? Charges have remained the same. Okay? Charges have remained the same. Charges remain the same, but ions exchange happen. Okay? So when I write about repolarization, I will write that efflux of potassium happened and potassium gated channels, right? Potassium gated channels opened. And that led to the ions exchange. Ions exchange. Clear to all of you? Clear to all of you? Here it is still positive. But instead of sodium, potassium has got opened in the repolarized situation. Okay. Now what is going to happen? In the next situation, they have to go back to the resting potential. They have to go back to the resting potential. That is when both the chains will open. Okay. Both the chains will open. See here. What is going to happen? Potassium will be taken in. Sodium also will be taken back to their places. Right? Yes. Great. Okay. They will go back to the resting potential when both the chains open, and they will go back to minus 70 MV. Minus 70 MV. Yes. Yes. This is one topic. Okay. We did we do good? Did all of you understand? Is there anyone who did not understand? Is there any student who did not understand? I don't think there's any student who did not understand, but I will label some parts for you people from NEET point of view. So students, I drew this gate, no? So this gate also has one name, okay? I'll tell you this. So this area, no, this top portion is called your activation gate. Activation gate. Okay? This down, the gate's down portion is called your inactivation gate. Okay? Inactivation gate. Clear? Thank you so much, Shara. Thank you so much. It makes me so happy to read such beautiful comments. So sweet of you people. I just want to make you people understand. That's all, okay? That's all is the goal. I do not care which technique I use for that. Even if I have to go down to an LKG student's level, I will do that. But my goal is to make each student who is watching this live worth their time and make them understand in the easy way possible. Okay? Yes. What hyperpolarization is here, Nancy. This is where hyperpolarization will happen. Here, no? Before going to the resting potential, they will go to a hyperpolarization state. That means both the gated channels will start, you know, hyperpolarization state will happen here. Okay, when both of them, both the channels, gated channels will start taking them back to their locations, and when that thing happens, hyperpolarization will happen. Thank you so much, YS. Okay. Permeability, actually your axolemma is more permeable to potassium and it is almost impermeable to your sodium. Okay, almost impermeable to your sodium. Okay, that also we'll write here. It is more permeable. Okay, that means your I'll draw a box here. Students, here I am writing whatever you people asked me. Okay. So, permeability, potassium, that is your axolemma. Axolemma is more permeable to potassium than sodium. Okay, than sodium ions. Students, don't forget to download this notes. It has everything. It has everything. Okay. Thank you, Lakshita. Yes. Yes, 45 to 65. Plus 45 to 65. I will explain you with the graph also, so it will be more clear to you people. Graph is clear? Oh, this graph, I should have this one. Okay, never mind. Students, so look here. I'll explain more about this in the graph also. So, minus 70 action potential. Okay, so let's write here also. So, minus 70, sorry, resting potential. This is the resting potential. Okay, resting potential. Minus 70 resting potential. That means fully blank. Okay. Suddenly a stimulus appeared, right? Suddenly a stimulus appeared. So, the stimulus is around minus 55 MV. That means it is capable of opening your gated channels. That means voltage gated channels. Now, can you see depolarization has happened. So, once the stimulus happened, what is going to happen? Depolarization is going to happen. Depolarization is going to happen. After depolarization, I told you it will keep increasing. Okay, during the depolarization, its goal is to cross 0. Right, from minus 55 threshold it reached, it is going to cross 0 and it will go up to 45 to 65. Some places you will see 45 to 50, it's actually 45 to 65. Okay, so after depolarization, action potential will happen in the plus 45 MV to 65 MV. Okay, that is this point. Okay, action potential. Clear? Right? Action potential is clear. Now, thank you, Lakshita. Okay, now students, with the action potential reached, it will come back to repolarization. That means your potassium channels are waiting to open. Sodium channels will close when action potential is reached. Right? Action potential. When we reach action potential, sodium gated channels will close. Okay, I am writing everything here. Sodium gated channels close. And what is going to happen? Repolarization, your potassium is waiting, right? Your potassium. So, here sodium channels are closing. Close. And here your potassium channels open. Channels open. Okay, potassium channels will open. After the potassium channels open, again here this one, you see this which you can see like this, it is showing, no, that is your hyperpolarization. This region is your hyperpolarization. Hyperpolarization. Okay. After hyperpolarization, again it will go to your resting potential. So this stage is where your potassium is going back to its place, sodium is going back to its place, because the voltage is reducing, right? Voltage is reducing, gated channels will close now. During gated channels closing, your extracellular fluid, your NP, that is your neoplasm, from all of that, whatever has come in will start going back, and it will come again to your resting potential. All the sodium in the extracellular fluid, all the potassium and negatively charged proteins in the inner side. Okay, negative in the inner side. No, action potential, see, action potential is 45 to 65. In some places it is written 45 to 50, but I believe it's 45 to 65. What is there anything mentioned in your NCERT? Wait, 45, 55, 45 to 50, 65 is written. Okay, 45 to 65 is written. Anything is written, students? Yes, no? Okay, yes. So this is your graph. Okay, so gated channels will close after action potential. After that, who is waiting? Sodium channels are waiting to open. Okay, at this stage, repolarization will happen. Repolarization will happen. Now, after repolarization, okay. Thank you, Sanvi. So, students, after repolarization, it will go to hyperpolarization. After hyperpolarization, resting potential. Students, after this, do not forget to read your NCERT. If you do not read your NCERT, it is going to be more difficult, because if you come later and try to understand or watch the video again, you will not understand, because now concept is very clear in your head. So, now as soon as the class gets over, take 5 minutes, read action potential at least from the chapter. Okay, very, very important. You should not miss your action potential, because it's a very, very confusing topic, because one were positive, one negative, so much is happening. So you should make sure that you are clear with it. So this graph is clear. Graph can come for exam. Something, if anything is missing, they can also ask for your board's exam. If you're a dropper, be prepared for your NEET exam as well. This can come. Okay. So 70, once again we'll go do a quick recap. 70 resting potential, stimulus happened, minus 55 it is going. See, can you see? Threshold, from there depolarization is happening. Depolarization means your sodium gated channels are opening because the voltage has reached for them to open. Sodium will start entering from your extracellular fluid, higher concentration to lower concentration, it will start entering inside. It will start making that electropositive, and outside it will become electronegative. Okay, after depolarization, once it reaches the maximum at 45 megavolt, that time action potential will happen. Okay, action potential will happen. After action potential reaches, sodium gated channels will close. Okay, sodium gated channels will close because of your inactivation gate. I showed you, no, that will close and the gates will close. Now once the gate close, so potassium is still waiting. No, potassium did not get its chance. Potassium gated channels will open. Okay, there is potassium gated channel, sodium gated channel, chloride gated channels, calcium gated channels. Okay, all these channels are there. At one point of time they will open. Okay, now action potential happened, then here potassium gated channels will start opening. So here what is going to happen? Repolarization, that means positive is going to be on the top, negative is going to be down, but exchange of ions have happened. That means potassium has gone out, sodium has come in, right? That potassium had made the positivity. Ion exchange has happened. Ion exchange is not okay. It has to go back to the resting potential. So during that time, here what is going to happen? Hyperpolarization will happen. Removing, taking everyone, your parents will come, no, keep quiet, don't eat my head, I'm already tired from work. So they will just do all separation, no? That separation will happen here, which is called hyperpolarization. Full hypertension your parents will be coming because you both fought. After that, you will all become calm for another one sometime. Until another stimulus happen, you will all keep calm. Okay, yes or no? Student, no, this graph is this graph has everything which is there in your NCERT. It's not an NCERT graph, but everything which is there in NCERT is here. Okay, nothing extra. Yes, it is a time voltage graph. Okay, yes, my God, we have done one big concept, right? I feel like I carried one mountain and came, and now it's broken down. Okay, students, surprise is going to be around after my class. Okay, after my class, maybe 10 minutes break. After 10 minutes break, you will have a surprise. Surprise, surprise. Yes. Okay, students, can you all see this? Okay. Students, here, this is what is mentioned in your textbook. I have just added, so my students, at least while downloading note, they'll be like, okay, let me open my eyes and read the textbook, right? So look here, when a stimulus is applied at a site, okay, stimulus has come. Students, all of this is happening in your axolemma, axon's outside part, okay. So when a stimulus has come on the polarized membrane, the membrane at the site A becomes freely permeable. So there is a diagram which is given where you can see A and B. Very confusing diagram, but still open your NCERT and stare at it, okay. So students, polarized membrane, the membrane at the site A, okay, becomes freely permeable to sodium. Freely permeable to sodium. That means your sodium gated channels have opened, because usually a membrane is more permeable to potassium but not very permeable to sodium. So here it becomes freely permeable, or in other ways you can tell your sodium channels open. Okay, this leads to a rapid influx of sodium. Okay, rapid influx of sodium, followed by a reversal of polarity. So reversal of polarity means positive, 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 positive was there, negative, 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 negative was there. It will become positive, positive, negative, positive, negative, negative, positive, positive, negative. Oh my god, so many negative and positive. Okay, now the outer surface of the membrane becomes negatively charged. Very important. Who will become? Outer surface. Okay, outer surface. Students, outer surface is called, it has ECF. Don't forget, extracellular fluid. Okay, negatively charged, and the inner side becomes positively charged. Yes, okay. The polarity of the membrane at the site A thus reversed and hence depolarized. So this situation is called depolarization. The electrical potential difference across the plasma membrane at the site A is called action potential. Okay. Now, which is the fact termed as your nerve impulse. That means one impulse has been made. At the site immediately ahead, the axon, that means next sign, okay. What is happening? Reversal is happening. Positive will become negative, negative will become positive. Again, outer surface and the negative charge. So the membrane has a positive charge on the outer surface and negative charge on the inner surface. Okay. Yes. I have. Then a current flows on the inner surface from site A to B. On outer surface, current flows from B to A. To complete the circuit, the polarity at the site is reversed here. Polarity at the site is reversed, and the action potential is generated at the site B. Thus, an impulse is formed. Now, here generated at site A arrives at the site B. My students, lot of A and B is happening. It's like a traffic signal. Even the vehicle person does not know where to go, even the traffic police does not know where to go. That is how much A and B they have written here. Okay, now the sequence is repeated along the length of the axon. That means, students, what is happening? Depolarization is happening, repolarization is happening, right? Then hyperpolarization is happening, then resting potential is coming. Then again D is happening, Re is happening, then RP is happening, right? Resting phase is happening. So D, Re, no. D, Re, RP, D, Re, RP. D, Re, RP is happening, students. I was trying to sing a song, but it does not happen. Okay. So yes, it is conducted. So stimulus induced permeability to Na is extremely short lived. It is quickly followed by a permeability of potassium, because they do not like, they do not like the, they know that younger ones always make problems. So they are more permeable to the elder ones. They are not very permeable to younger ones. So immediately they will come back to their, this one. Okay. Yes, that's all. Students, that is what your NCERT, if you read somewhere, you will understand. Okay. But with the diagram you have to read, otherwise you are not going to understand. Okay, now we told so much, no? Full story I have told you. Storytelling is over. I'll give you a break, okay? I'll give you a break. I'll give you a break before starting brain, so that my brain also calms down, because full neuron is there, full action potential is happening. My brain will be like, why are you putting so much pressure on me? Power, I've never worked so hard. Okay, so my brain is, I'll take a break for my brain and also give you some break. Okay, okay. Students, look here, myelinated neuron, very important concept. Okay, so this, let's just draw very simple neuron. How many of you like brain concept? Okay, students, this is a simple axon. Okay, I'm going to draw myelin sheath very big so all my students can see. Yes, done. Okay, simple axon. This looks like a tree, but it's not a tree. Okay, this is an axon. Now students, we understood action potential. We understood that from one place to another place to another place it's going. Okay, simple. Students, I didn't ask who all have brain. I asked who all like brain concept. Okay, who all like brain concept. Okay, don't tell ma'am I have brain, you also have brain. Okay, do not say that. I asked who like brain concept. Okay, students, this is myelin sheath, correct? That means your Schwann cells will start forming myelin sheath here, right? Myelin sheath here will be formed. Now, when action potential is happening, so say here it is happening. Okay, here plus minus is happening, here action potential is happening. Now from here, okay, from here it will jump here. That means in wherever myelin sheath is there action potential is not necessary, correct? Okay, action potential is not necessary. So from here, from that gap, from the node of Ranvir, you know, from there to a next one, from there to the next one, from there to the next one. So when the axon is myelinated, that means when the axon is myelinated, okay, when we have myelinated neuron, the type of conduction is called as saltatory conduction. Saltatory conduction. What do I mean by saltatory conduction? They will jump. They will jump from one place to another instead of walking. Okay. So jumping is happening. Now students, it's your wish if you want to jump and go to the kitchen or walk and go to the kitchen, right? If you jump, your mom will end up calling you monkey. If you walk, your mom will be like, walk more slow so you don't have to watch ma'am's class, right? So whatever you do, you will still get scoldings, but that is different. But for neuron, it's not like that. If it is jumping, we will appreciate it. Why? Because when a normal, say a normal axon is there, students, normal axon, okay, normal axon is there, okay. I ended up drawing like a frog's leg now because it's too difficult to draw fast. This is a non-myelinated axon. Okay, here what is happening? Action potential has to keep happening, correct? Keep happening, keep happening. If it has to keep happening, that means your potassium and your sodium pump also has to keep pumping, right? Pump also has to keep pumping. When they have to pump, what did I tell you they need? They need ATP, right? They need ATP. When this happens, continuous ATP is given out, right? Continuous ATP is used up, okay? But here you see, here they need only one ATP, here they need one ATP, after that here they need one ATP. Here it's not like that. ATP, 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 every single time they need ATP. So saltatory conduction or myelinated neuron actually is more efficient because we require only less ATP. Less ATP is required. Clear? Less ATP is required. Clear? Hey, this is not root. One person is asking me, ma'am, why root should jump? I will jump and go out of the class now at this point. What and all you people ask me, this is axon terminal. I knew one or the other one will be there to ask. All one species you people are by itself, no? Right? Students, here, node of Ranvir. Why you people like Ranvir so much? Huh? That I didn't mark so you will not learn. So here, this one, where to draw? No space only. This one. Node of Ranvir. Node of Ranvir is there between your both myelin sheath. Okay. Students, this is clear. So question can come, why saltatory conduction is better? You will say less ATP is used, more efficient, because neurons or the action potential jumps. So the amount of ATP that is required for pumping is less. Okay. Right? Yes, total emotional damage has happened. Your own jokes help you or my jokes help you? Ah, very good. He corrected it. Ritesh understood that this ma'am is going to attack me in 1 second. So, very fast I will make ours into your. So, ma'am does not attack. He added Y faster than his life. Like speed of light we tell, no? I don't think he would have even answered mock test so fast. He's like, Y he made of. Copy pasted Y. Okay. So, students, this is clear. Myelinated sheet is clear. Okay. This question can come. Saltatory conduction. Saltatory conduction. Okay. Yes, that's all. We are done with neuron. Graph done. I'm looking at your NCERT also. Graph done. Stimulus done. Now we have only brain. Yes or no? Brain, only brain. Brain is there. Oh, no, no, no. Shusho. Chemical synapse. Forgot, students. Sorry. Chemical synapse we have. Okay, shall we take a break and come back? I'll give you 7 minutes break. Fine. Don't run away. Don't run away. I want all this 57 students to come back. Break? Yes. We'll finish off synapse, brain together till 8:30, no break. Okay? Because we have launch today. We have, not launch, surprise. Go drink coffee, recharge your brain and come, because this portion is very easy. Synapse is very easy. Yes, all of you should be back, okay? Yeah, break. Students, only 5 minutes. Run and come back. Just so we can drink water and come back. Yes? Other organs, we don't have Praveen. Portions are deleted. We only have brain. We have only brain, no sense organ, no reflex action. Okay? No reflex action. Please do not break your brains. It's not needed. Okay? It's not needed. Right? Okay, go for a break. Bye. See you all at 6:40. Yes, come back. All of you should come back. Okay? Otherwise, no brain. Students, are you all back? I am back. Are you all back? Are you all back? Yes. Wow. Good job, good job. All of them are not back. Let all of them be back. By then I'll draw synapse. Right? Let all of them come back. By then I'll draw synapse. Yes, all of you are back? No, let me tell them. Let me tell them. Let me tell them. Then they'll be back. Breakdown, join. Yes. Most of you are back? Yes, back. Okay, students, so by then I just drew just synaptic, little bit I drew. Okay. So now we are done with electric transmission, right? We already understood that when the neurons are completely coming, right? When there is no gap, electric transmission happens. When there is a gap, chemical transmission happens. So chemical transmission happens because of the presence of synapse or your synaptic bulbs which are present in the axon terminal. All of you know all of this basic. Students, all of you are not back. This is the reason why I don't give breaks. Okay, why I don't give breaks. Yes, definitely. Dedicated doctor, if you are very dedicated, you will definitely get more than 500 if you start following us now. For your physics, chemistry as well as bio, even you will get 600 above also. Okay. In bio, pakka you will get 300 plus. But you have to be dedicated. Like your name says, you have to be dedicated. Okay. Yes. So students, chemical transmission is happening because of your synapse, and this synapse or your synaptic cleft, synaptic nerve, all of this are present in the axon terminal, right? Axon ending. Is there any student who did not see the neuron diagram? The beautiful diagram, how can you not see? Students, look here. This is your axon terminal. From that, the bulb is the synaptic bulb which I drew in the large. Inside this, you have synaptic vesicle. Inside that, neurotransmitters are there. So now we are only going to draw synaptic vesicle in our this diagram. Okay? Clear? Where is it? Yes. Okay. Students, so what are we going to understand? So synaptic cleft, I told you if the gap is more than 2 meters, right? I told you if the gap is more than 2 nanometers, then we will do chemical synapse, right? We will do chemical transmission or chemical synapse, both words you can use. Okay? Yes. So inside the synaptic vesicle, okay, inside synaptic vesicle, we have neurotransmitter. Okay, neurotransmitters. Okay, neurotransmitters can be excitatory, that means they can excite or they can inhibit. Okay, So neurotransmitters can be of two types, excitatory or inhibitory. Okay, that means they can excite a impulse or excite a reaction, or they can inhibit it. Okay, or they can inhibit. Okay, so this much is clear. Now let's try to understand. Okay. Yes, give your dedication, then we will be good to go, right? Good to go. So students, chemical synapse is very, very beneficial for us mainly because energy consumption does not take place. Electrical transmission, ATP is continuously needed for the pumping to happen, but here that is not needed, right? ATP is not needed, so this is more beneficial. Okay, this is more beneficial. Yes. Okay, so excitatory, inhibitory. Now let's try to understand how this chemical transmission happens. So students, in this whole thing will be divided into presynaptic, okay, presynaptic, postsynaptic, postsynaptic, and do you see this gap here? This gap is called a synaptic cleft. Synaptic cleft. Okay, CLEFT, synaptic cleft. Now, at the end of your synaptic knob, there are neurotransmitters, right? Neurotransmitters are stored inside your synaptic vesicle, right? So, neurotransmitters are there, neurotransmitters are there, neurotransmitters are there. Okay, so as soon as an impulse reaches from the dendrite to the cell body to this axon, to the axon terminal, what is going to happen? This synaptic vesicle will come and attach itself here. Okay, so I am erasing here and I am drawing it here. It will come and attach itself here. Okay, it will come and attach itself here on the edge. Inside this, again, neurotransmitters will be there. Okay, after some time, this will break open. Okay, this will break open. Can you see this? This whole thing will break open. Why? Neurotransmitter has to come out, right? Neurotransmitter has to come out. Students, yes, synaptic cleft. So this portion is your synaptic cleft. You can also call it synaptic gap. Gap also you can call it, or synaptic cleft. Okay. So students, here this one, this synaptic vesicle will break open and your neurotransmitters will be transferred. Okay, neurotransmitter will be transferred. Any doubts in this? Easy? So how is this happening? I told you, when I explained you gated channels, I told you there is chloride gated channels, calcium gated channels, potassium and sodium. So the moment this happening, that means impulse has to happen, the calcium influx will happen. Okay. When the calcium influx happen is when this synaptic vesicle will go attach and this neurotransmitter will open. I mean, the synaptic vesicle will open and the neurotransmitters will be going out. Okay. Yes, easy concept. You have studied this. Yes, easy peasy lemon squeezy. Right. Okay. Yes. Now students, neurotransmitters are just scattered like this. Will they just be like that? No. For them, there will be receptors here. Okay, receptors will be there. On the postsynaptic cleft, receptors will be waiting to get this neurotransmitter. They will be waiting for this acetylcholine or dopamine or for other things, right? For them, glutamate, all these neurotransmitters to come and bind on this receptor. So this receptor will collect or take the neurotransmitter. That means what is happening? Information is getting passed. So neurotransmitters will make sure that information is not leaked and every single information is going to the postsynaptic cleft. Easy? Very easy, no? Yes. Yes, synapse requires stimulus and synapse requires calcium influx. Okay, calcium influx has to happen, otherwise this thing will not happen. Okay, calcium influx has to happen. Okay, is there any doubt so far? No, right? Easy? Easy? Okay, so that is all about your synaptic cleft. There is no much explanation, but I have also added a diagram for you people to understand, because in case I draw it and you don't feel like, oh ma'am, I don't understand it. Students, you can see a small portion here. So can you see here? How is the connection? I told you head of one, tail of one, head of one, tail of one, right? So look here, this is head of one, tail of one. So axon terminal is getting connected to your dendrite, right? So this becomes your dendrite, your post-synaptic neuron. This becomes your axon terminal. So here you can see there are synaptic vesicles inside the synaptic vesicle. So here if you see, there are voltage gated channels for calcium. So calcium influx will happen because of voltage gated channels, okay? And then as soon as calcium enters, these things will go and bind to the end, at the end of this synaptic cleft, right, near the synaptic cleft, and then the neurotransmitters will start coming out, and these receptors will be waiting, okay? Ion channel receptors will be waiting to accept the neurotransmitters, so the impulse can be passed from gaps, Clear? Very good diagram. See, look here. Here this portion they have zoomed in. In your notes it will be more clear, okay? In the notes it will be more clear. Yes, done. Okay students, now small information, okay, small information about your inhibitory and your excitatory neurotransmitter. After that we will be done with this portion, okay? So when I talk about neurotransmitter, just write this, okay, write this all of you, important from your, there are so many very dedicated students after the break just does not come back. This is what we call them as false dedication. Yes ma'am, I will come. Ma'am, whatever happens, I will come after the break. Even if earthquake happens, I will come. They won't come. These are the students who are in the class right now. The 39 of them who are watching the live right now are the ones who are dedicated and are the ones who are going to be in the most prestigious colleges, that is going to, after May 5th. Okay, once the results are out, these are the names that we all as teachers will be waiting to see, and as students you people should be eager that yes, I have dedicated my time to this teacher and I trust her, and I will definitely be in the colleges that I want. Okay, so students, every time we say this, we are not telling out of telling that oh, more people come to our live or make us famous, not for that, but it is a blessing. Like whenever we see your names, we feel happy. Yes, one day if my one student does not come, you people won't believe, I even text them on Telegram and ask like, are you all okay? Is there anything that you have faced problems? I do that. Sometimes I do that because they have been there with me from the starting of my journey till this date. Okay, till this date they were with me, so I know them. I care for them. I care if they are attending, if anything is wrong, if I have to mentor them in any way. That kind of dedication you will see from my side also. Okay, so it is give and take policy, right? Every action has an equal and opposite reaction, Newton's law. So much physics I know. Okay, so neurotransmitters. So be consistent, and watching live will not make you lose anything. Okay, yes. So students, excitatory, okay, okay, and inhibitory. Let's see who are the neurotransmitters that inhibits. Okay, inhibitory. Yes. Thank you so much. Okay, this excitatory ones helps in depolarization. So definitely this person will help for repolarization. Yes. Now examples. Let's see the examples. Okay. So which is the first one I told? Very common one which you have studied, students. Yes, I know. Thank you so much. Yes, even I will be there for your journey till the end of your journey also. Right. Acetylcholine. Okay. Glutamate. Okay. Here who and all are there? Dopamine. So these are some excitatory ones that will help for depolarization, some for repolarization. Okay, then we have GABA, Okay, GABA, glutamate, serotonin will come here, inhibitory. Yes, all with me? Dopamine, glutamate, very good. Epinephrine, yes, epinephrine will come in excitatory. Okay, yes, this is your examples. Now I'll tell you few more things which I'll write in the middle because there's no place. Okay, so this will go in the middle. This is the first time anyone will write like the students. Okay, Gopika ma'am is different, no? So I will also write differently. So I will just write what happens in case low concentration comes. Okay, low concentration comes. So low concentration of what? Let's do first. We'll do acetylcholine. If it is low acetylcholine will lead to Alzheimer's. Okay, Alzheimer's. Students, Alzheimer's you have seen in that Rocky and Rani movie. Rocky and Rani, they will say no. They'll say about their father. He forgot, right? He lost his memory. Because of that, he's not able to remember, and then they'll bring the heroine. All of you remember? I don't know how many of you watch Bollywood movies. So Alzheimer's, many movies they have shown. Okay, so low acetylcholine will lead to Alzheimer's. Then your dopamine is there, low dopamine. Okay, low dopamine, Parkinson's. Okay, Parkinson's. Students, Parkinson's. So what happens is when low dopamine is there, have you seen some people, they cannot control the movement. Their hands will be always moving, right? Have you noticed? They cannot stop. Dopamine does the job of inhibition. So they will stop when they're doing something. But if dopamine is not there, they will continuously keep moving. If you have seen some old people, they'll keep moving. They cannot take anything. Both the hands will shiver and it will drop out of their hand, right? Parkinson's. Okay, Parkinson's. Okay, that is your low dopamine. Okay, clear to all of you? Yes, Shivani, don't worry. At the end of this class, this chapter also will be in your fingertip and you will attend every other class. Okay, yes or no? Done? Yes, very good. Okay, students, this is all about your neurotransmitters. Now we will study brain. Oh my God, brain. Ma'am, brain. So students, I told you before brain, I actually wanted to teach you nervous system. One more part. So let me just take one empty slide. Okay, let me take one empty slide and explain you nervous system. Going faster than aeroplane. Yes, nervous system. Students, nervous system is actually given in the starting of your this one, but I am explaining it now. Okay. Students, by the way, don't miss the surprise. All the teachers are in the office. All the teachers are in Vedantu office. Okay, and yes, don't worry, Shaina, the team will do it. Okay, we are doing it for you people, so don't worry. Ho jayega, right? Ho jayega. Okay, students, look here. Nervous system is divided into simple CNS, PNS. Central nervous system, peripheral nervous system. Now, who does not know this? Everyone knows this. Okay, central nervous system is again divided into brain and spinal cord. Do I need to explain this? Brain and spinal cord. Yes or no? Easy? Okay. Now, PNS, students, we have always I don't know if whenever you have learned from teachers also in school, they always tell you that brain and spinal cord is like the center. See, now if you want to come to Majestic, okay, that's one place in Bangalore. Imagine you want to come to Majestic. If I want to come to Majestic, there are 100 routes to come to Majestic. Or from 100 routes also people will be coming to the central place. How are people coming? Different transport, bike, car, metro, bus, all different transport they are coming. Train, all this different transport, right? They should be long, they should be efficient, they should be able to bring you from one place to another place. Same way, PNS does the job of bringing whatever signals and nerves that is required for your central nervous system. Okay, so PNS has your cranial nerves, okay, that means the nerves that come to the brain, and spinal nerves. Definitely cranial nerves will be lesser than spinal nerves because spinal cord is longer, correct? Spinal cord is longer. So cranial nerves are 12 pairs and this is 31 pairs. Lahari, I'm seeing you after so many days. Where were you? I thought you went missing. Where were you? I mean missing from my class, okay, not missing from my class. Okay, so 12 pairs, 31 pairs. Okay, this much clear. Now students, PNS or your peripheral nervous system can be again divided depending on who will do voluntary, who will do involuntary. That means my hands, voluntary. Please don't take this and put that ma'am is dancing in the class. Okay, okay. So voluntary. Legs, voluntary, okay, involuntary, smooth muscles, okay, glands, all that involuntary, okay. Now whichever is going to do voluntary, that means whichever is going to take this impulse from the CNS to the voluntary part, okay, voluntary part, they will be, okay, can you understand this? Now 1 second, I'll draw with, students. Okay, this is not how I plan to write it. 1 second again. Okay, students, so PNS will be again differentiated into two parts. Okay, somatic neural system, okay, and autonomic neural system. Okay, now what did I say? I told whoever is doing the involuntary, okay, right, somatic neural system, autonomic neural system. That means somatic will take impulse from the CNS and pass it on to voluntary part or your skeletal muscles, according to your NCERT, skeletal muscles. Okay, again autonomic nervous system will take your impulse from the CNS and pass it on to your smooth muscles. Again NCERT, smooth muscles, glands, etc. Okay, clear? Okay, this is some dam opening types of separation. Okay, this is not how I planned it would to happen, but it just happened. Okay, so students, somatic neural system and your autonomic neural system, you can call it SNS and ANS. Okay, answer ANS. Yes, now that is clear. Now your autonomic neural system is again divided, but I don't know where I'll divide. Okay, again it is divided into sympathetic and parasympathetic. Okay, sympathetic and parasympathetic. Yes, every problem has a solution. Okay, are you all with me on this for my problem and solving solutions? Yes, okay. So students, autonomic neural system will have two more differentiation, para and sympathetic. Okay, sympathetic and parasympathetic. So sympathetic is during emergency situation and parasympathetic is during rest situation. That means when you are relaxed. Okay, parasympathetic. Do not forget to download the notes. There is a login option given. That means you enter your number and OTP so that we are making sure that all of you can also write on the notes, mark on the notes, right? Like see, now I have written, but you are feeling like, okay ma'am, I need to also write about the spinal cord something, but I have not written. So you can log in and you can use that tool to write your own notes on my notes personally for yourself. So don't be like, who will enter the phone number, OTP will come, who will look at the OTP, who will enter, my god, so much brain I cannot use. you will know how complicated your brain is. You can definitely do multitasking and many more work. Okay, so do not forget to download this notes. Which tree? This one. I'll tell you which tree this is. Christmas tree. Okay, Christmas tree. Happy? Oxytocin. Arbor vitae. Okay, students, clear? This is the differentiation of your nervous system. Easy. Sympathetic, parasympathetic, somatic, autonomic, cranial, spinal, brain, spinal cord. Now brain you are going to study. Okay, brain is divided into which and all parts, students? Simple. Brain is divided into forebrain, midbrain, hindbrain, okay. Brain is divided into forebrain, midbrain and hindbrain. Okay, this much done. Okay, now wait, now I'll go back to brain. Yes, let's come to brain. Students, brain, forebrain, midbrain, hindbrain, already I told. Yes, forebrain, midbrain, hindbrain. Clear? Okay, now let's start from the starting. Students, brain is such an important part that without that you are not even called a human. Why? Because animals also have brain. Animals also have some kind of thinking. There is ganglions in smaller vertebrates, right? So many things are there. But why are they not able to think like us? I am able to stand in front of you, camera is there, mic is there, studio is there, everything, because man was able to do it, right? So our brain has to be protected more than you protect anything else. Like how you carry your phone, like this you carry, no, you don't fall down, don't get up, don't, you'll keep it on the bed like as if it's one alive baby. You'll keep it on the bed, then you'll take it from the bed, and in case you bought an iPhone very recently or something, then gone, you'll carry it on your head and walk around, right? Same way we have to protect our brain. Okay, we have to protect our brain. So brain is protected by a bony structure. Bony structure. What is the bony structure called? Cranium. Cranium. And can anyone tell me cranium is made up of how many bones? 8 bones. Okay, I only ask the question, I'll only answer, students. This is self-answering. Okay, because you people are very slow. Okay, cranium. How many bones? 8 bones. Okay, 8 bones. In that, 2 are paired, the rest are unpaired, right? 2 are paired, rest are unpaired. You will study more about this in locomotion and movement. Okay, so cranium, brain box where the brain is kept, so nothing happens. Okay, students, you will get the notes today only. At the end of the session you will get the notes. Okay, yes, 8, very good, 8, good job. Okay, now let's just draw a very simple brain, just to understand, not to make it complicated, just to understand. Let's draw a brain. Oh, very good. Now it's fast. Now you people are fast and furious. Okay, okay, this is brain. Let's draw a helmet. That is your cranium. Okay, cranium. Then let's draw meninges. Yes. Okay, students, meninges also done. Okay, now we studied cranium. So this green color part is your helmet, right? Cranium. Then after cranium or below the cranium, there's something called as meninges. Meninges are divided into 3 parts. Okay, meninges are divided into 3 parts. Okay, first one is your dura mater. Okay, so one second, I have to draw more of this meninges. Okay, done. What did I say? After cranium there is meninges. So meninges are of 3 layers. Okay, meninges are of 3 layers. They are also again for protection, right? Meninges are of 3 layers. Okay, first one is dura mater. Then arachnoid and pi matter. Right? All of you are with me on this? Yes, easy? Can't see any reply. Okay, arachnoid, dura matter, this one. Okay, students, so here when we start layer wise, say, okay, dura matter. Okay, wow, helmet only went off. Students, 1 second, yeah. Then we have, okay, dura matter is there. Then we have arachnoid. Okay, then the pi matter is actually the part of the brain itself. Okay, so you cannot differentiate it. Okay, pi matter. Now, between the dura matter and the arachnoid matter, we have a sub. Okay, there is a space called as sub. Okay, so the space, very lazy. I'm very lazy, ma'am, it seems. Students, 1 second. Yeah, students. So between the dura matter and the arachnoid, there is a subdural space, okay, which is filled with blood vessels, okay. I don't like this diagram, so I'll erase it after this before your notes is given, okay. And between the arachnoid and the pi matter, there is a space. So what will that be called? If this is called subdural space, this will be called subarachnoid space. Easy, right? Subarachnoid space. What is present in this? Very, very important. Your that is your cerebrospinal fluid. You have all studied this in your 10th and all, but you didn't know where it was present. Cerebrospinal fluid is present in the subarachnoid space. Okay, cerebrospinal fluid acts like a shock absorber. Okay. That is how your brain is still there in place. Otherwise, how many times in small age you would have fallen, gone and hit on the bench, desk, tap. You would have opened the tap like this and you would have lifted your head like this. Okay. So, your brain, nothing has happened is mainly because of your cerebrospinal fluid. That means any jerk happens, no? That shock absorption is done by your cerebrospinal fluid. Okay. Yes. So, if I am, so students, this much is clear. So, if I am going from inside to outside, that means from inside to outside, it is called pad. Pad. What is the pad meaning? Meninges, order of meninges. Inside to outside, pad. Outside to inside, dap. Okay. Inside to outside, pad. Outside to inside, dap. Dap, pad. Dap, pad. Okay, clear? Yes or no? Any doubts so far? Let me know now. Any doubts so far? Okay. Students, this cerebrospinal fluid is also present inside. Okay. Inside this also there is going to be some space. Okay. Some ventricles. Okay. Ventricles are there. Okay. Here also your CSF will be present. It will go all the way outside. Okay. CFS will be present. Yes. This is clear? Yes or no? This is the basic. Okay. Pad is inside to outside. Pi matter, arachnoid, dura matter. Outside to inside. Dura matter, arachnoid, pi matter. Pi matter is in your brain portion itself. So, you cannot remove the layers and see. But if you see dura matter and arachnoid, they will look like a fluid layer. It will look like a fluid layer. Okay. If you put a plastic cover around a pink sponge, how it will be? One plastic cover, one more plastic cover. So, first plastic cover will be dura matter, second layer will be a arachnoid, but pi matter is not like that. It will be on the pink sponge. Clear? Okay? Yes. Great. Now, brain is already divided into forebrain, midbrain and hindbrain. So, we are just going to write what is present in each of the layers. Okay? Forebrain. Okay? Yes. So, first forebrain. Okay. Forebrain. Students, what and all forebrain is present? Can anyone say forebrain? First, largest part of the brain. What is it? Cerebrum. Right? Largest part of the brain, cerebrum. right? Second, diencephalon. All of you forgets this. Diencephalon. Last one. Last one, can anyone say? Come on, let's give it a try. Last one, what? Olfactory. Very good. Olfactory lobes, right? Olfactory lobes. Yeah, olfactory lobes. Okay, let's also write about diencephalon's classification. Students, diencephalon has three parts: epithalamus, okay, thalamus and hypothalamus. Okay, hypothalamus. Clear? Yes, this is your right. Cerebrum, diencephalon, olfactory lobes, epithalamus, thalamus, hypothalamus. Okay. Now in midbrain, okay, students, I'll do one thing. I'll write this side. Me and my space problems. Right. Diencephalon, let's write it here, students. Okay. What did I say? Epithalamus, thalamus, okay, and hypothalamus. You will study more detail about hypothalamus in your endocrine. Okay, midbrain. Okay, midbrain has two parts. Yes, all of you are with me on this? Any doubts so far? No, right? Corpora quadrigemina. Okay, and first one is corpora quadrigemini. Very good. Second part is cerebral peduncle. Cerebral, whose uncle? Peduncle. Okay, got cerebral peduncle. Then last part, midbrain. Oh, sorry, hindbrain. Okay, hindbrain. Which are the three parts? Cerebellum, second largest part. Pons, where are they? Medulla oblongata. Medulla oblongata will go into the spinal cord. Yes or no? Done? Done? Yes? Okay. One second, students, one second. Yes? Okay. Yes, very good. Clear? So this is the three parts. Okay, cerebellum, pons, medulla, it will go to spinal cord, corpora quadrigemina, cerebral peduncle, cerebrum, diencephalon, olfactory, epithalamus, thalamus, hypothalamus. Yes, yes, yes. So this is the same thing which we just did. Okay, so brain is the central information processing organ of our body and it does the command and control system. Okay, and it controls voluntary movement, balance of the body, functioning of the vital, involuntary and voluntary organs, thermoregulation, hunger, thirst, everything. Everything is done by your cerebrum, right? Your first part. So here, students, thermoregulation is done, hunger is done, thirst is done. Okay, circadian rhythms. That is the reason why if you one day sleep at 4 o'clock, next day again you have a tendency to sleep at 4 o'clock. You are not able to go back to your original original timing is because of this circadian rhythm. Okay, activities of several endocrine and human behavior. All this is done by your brain. Okay, that is why your brain, no, sometimes you will be like, should I like this session? No need, I shouldn't like the session. Should I share it with my friends? No need, I won't share it with my friends. That is all done by your brain. Okay, that's all done by your brain. Okay, so it is also a site for processing of vision, hearing, speech, everything, right? Now the human brain is well protected by a skull and it also has, inside the skull, it also has cranial meninges which are divided into three parts, DAP, right? using an outer layer called the dura mater, a very thin middle layer called the arachnoid, and an inner layer which is connected with the brain tissue. That is why I told you, you cannot remove it and show, right? When you see the 3D videos, you see that pi matter cannot be removed and showed. Okay? Yes? Yes, I will come to that. Hippocampus and all that. They are your emotional. Sharath, you cry? No. You cry. You get very happy. If you crack your NEET, how happiness, all that is controlled by these people. Okay? The brain can be divided into major parts: forebrain, midbrain and hindbrain. We are already done with this, right? So this is a diagram just to show you people. You can refer it through the notes. Okay? Yes. Students, forebrain. Let's start forebrain. All the parts of your brain is working? Yes, no? It's working? Okay. Students, so forebrain, you know that there is a frontal lobe, occipital lobe, parietal lobe, all that you know, no? Temporal lobe, all of them are separated by your sulci, okay? Sulci separates it, different types of sulci are present. Now, when you talk about forebrain, the first thing that you talk about is your cerebrum. Yes or no? Cerebrum. Yes? No? No ma'am, I don't talk about brain only. So before that, I will just write it in one corner. This question was asked in previous year. So that is why we will just mention it here, students. Brain stem. Okay. Brain stem consists of what? Students, brain stem consists of MPM. Okay, not just MP, MPM. That means M stands for midbrain. Okay. P stands for pons. Another M stands for medulla. These three people form the brain stem. This question was asked. That is the reason I am mentioning it here. So make a note of this. It will be there in your notes as well. Brain stem. What does brain stem comprises of? Brain stem comprises of MPM. MPM. Midbrain, pons, medulla. MPM. Okay. It's working, ma'am. Okay. Okay, students. So when we talk about that, you know the frontal lobe, occipital, parietal lobes and all. I'm not drawing that, but you know that. Okay, I will send a picture of my notes as well, maybe. Okay, so in case I will draw one, I'll just draw. Okay, so I'm just going to draw just the outer surface just to show you the parts. Okay. Okay, so students, this does not look like a brain, but please adjust. This is a brain. Okay, I'm telling you, so you should believe that this is a brain. So here you have your frontal lobes. This is just to show you the lobes. Okay, here we will have parietal lobes. This one, this portion. Here you will have your temporal. Okay, can you see this much? Yes, here temporal. Got it? Now this portion has to be more attached. 1 second. Okay. Yes. Student, this is just to show. Okay, this is just to show your parts. Okay, I am only laughing at my own diagram, but it is fine. It is manageable. It is for understanding. So yes, let's just label the lobes. Okay. Thank you, Soumya. Let's just label the lobe. Frontal. Okay, frontal. And this frontal. Okay, frontal lobe. This portion is the frontal lobe. This is your parietal. Okay, what will be this? What will be this? Occipital, right? Occipital. This is temporal. These are all separated. This, no, what you see is your sulci, right? Sulci. It is separated by your sulci. This too will be separated by your central sulci. Central sulci. Okay, parietal. So many parts are there. Yes, correct. Okay. Frontal, parietal, occipital, temporal. Yes or no? So this will be your parietal, occipital, sulci, right? This will be your parietal, occipital, sulci. So this is more of your understanding. That is why I am not putting a lot of pressure on this. They won't ask you, or they'll just ask you the lobe's name. They will not ask you to draw and show. Okay? Parietal, occipital, sulci. Okay, clear? Fine. Now let's understand cerebrum. So cerebrum, what and all do we know about cerebrum? It's the largest part, right? So cerebrum is the largest part of the brain. Largest part. Now this largest part has two parts, right? There is a left and a right hemisphere. This two, left and right, we call it as what? Cerebral hemisphere. Cerebral hemisphere. This is all basic, so I'm going little fast. Okay, so cerebrum. Gopika ma'am's diagram is better than NCERT, it seems. Students, I'll cry. I'll sit and cry in one corner. Why do you people tell me such things? Don't motivate me to do such things. Okay, so this is for your understanding, right? We cannot draw brain and all so easily and so smoothly and all that, right? We are not artists, we are teachers. So our main concept is to make you learn properly. So students, we have left and right hemispheres. It is called as cerebral hemispheres. Okay. Now, this, when we talk about your cerebral hemispheres, all of you remember in your 8th standard and in your 10th standard, there was a chapter called life process and there is reflex action. 10th standard reflex action, if you remember, there's a concept. Most of the students find it very difficult. In that, you would have seen the spinal cord. There is a H-shaped structure, white matter, grey matter. Remember? White matter, grey matter. Yes or no? So, here also cerebral hemispheres have an outer and an inner region. Okay. This outer region, okay, this outer region will have the grey matter, that means the regions wherever the myelin sheath is not present. So tell me the regions where myelin sheath is not present. Let's see our neuron once, then you will understand. Tell me where and all myelin sheath is not present. Myelin sheath is not present in the cell body and it's not present in the axon terminal, right? Cell body, dendrite. Every place myelin sheath is present. So this grey matter is made up of what and all, students? One second. Yes. Grey matter will be made up of what? Your cyton and axon terminal. Okay. Grey matter is made up of your cyton plus your axon terminal. So the outer region of your cerebral hemisphere has the grey matter and your inner region, right? Inner region will have your white matter. Inner region will have your white matter. Okay, one second. This will have white matter. White matter is a region because we told myelin sheath, right? White color. So white matter is the region where myelin sheath is there and that is axon. Simple, easy. Easy this much? Easy, right? Yes or no, easy? So students, this, if you have noticed, you would have seen brain structure, you would have seen something like this, right? All of you would have seen something like this, right? Brain full, you can see like this structure, right? This structure you can see. So that is your cerebral cortex or your cerebral cortex layer has complete grooves. Yes or no? All of you have seen these grooves. You wouldn't have seen a brain which looks like a helmet, clear Vega helmet, pink color. No, you will see so many grooves inside. That is to make more neurons come in. They say, no, people who have more neurons are more intelligent. Like that. More, to increase the surface area, such grooves are present. Okay, such grooves are present. This top portion, okay, here, this top portion of the groove is called as gyri. Okay, gyri. And this down portion, which is coming downwards, right? This down portion is called your sulci. Sulci. Got it, all of you? Yes or no? Cerebral cortex have grooves together. This gyri and sulci together are called grooves, right? If the groove is very long, we call it fissure. If the groove is very long, say imagine last one groove, I draw it very long. Okay, like this side too. Okay, that means more surface area. This is called a fissure. This is called a fissure. So gyri is the top portion, sulci is the down portion. Okay, small one we call sulci, larger one we call fissure. Okay, S stands for small, so you can think of that. Yes or no? Yes or no? Clear? Clear? This much is clear? Any doubt so far? I don't think there is a doubt. Doubt is there? No, right? Okay, so Gairi and sulci clear. Now students, here one thing is mentioned in your NCERT. Let's just go through that. So here the forebrain has cerebral, thalamus, hypothalamus, not required now. We have discussed this. See here, this line, okay, come to this line. So the layer, okay, 1 second. Students, this here one more thing we'll write off. Which color to choose? Yes. Students, your right and your cerebral hemispheres are joined together because of corpus callosum. Right? Your right and left hemispheres are fused or joined with the help of corpus callosum. Okay, that is what your first line is here. Okay, now look here. Two halves are joined. Okay, they are called as cerebral hemispheres. They are joined by corpus callosum. Now look here. The cerebral cortex is referred to as the grey matter due to the greyish appearance, presence of what? Presence of your axon terminal and cyton. Okay, I am writing C and axon terminal, AT. Okay, cat. Okay, the neuron cell bodies are concentrated here, giving the color. Okay, now cerebral cortex contains motor areas, sensory areas and large regions that are neither clearly sensory nor motor in function, and they are called as association areas. So very, very important. So I'll tell you about the neurons, do not worry that, but all of you know that there is sensory neuron and motor neuron. That means one will take information from the sense organ to the CNS, right? Sense organ to the CNS. That person becomes a sensory neuron. Now one person takes the information from the CNS to the effector. That becomes the motor neuron. Between that, there is something called as your association neuron, relay neuron. You would have heard many names. Intersensory neuron. All of you know this? Intersensory neuron. All this is what mentioned here. Now, this association neuron has the ability for memory, communication and intersensory. Intersensory. They are not completely sensory. They are not completely motor. So, they become intersensory. Clear to all of you? So, they have complex functions like intersensory association, memory and communication. Very, very important. Very, very important from NEET point of view. Okay. What is happening? What is happening? Okay. Shaina, Sharat, I will explain. Students, fissures also increase the surface area. Deeps. Fissures also increase the surface area. More the surface area, more the neurons can fit in, right? So, that is, okay. Students, do not spam. 1 minute if I look this side, no, they will start spamming. They will talk to themselves. Did you eat right food? How is life? Fibers of the tracts are covered with myelin sheet which constitute the inner part. That is your white matter. Okay, it constitutes the white matter. Is this association neuron clear? Association neuron clear? Yes, no? How will you understand if you are talking to yourself? You will not understand if you are talking to yourself. So, students, here, let's write it in a box. Okay, let's write it in a box here. So, what is the box going to tell about? So, there are two types of neurons, right? Two types of neurons. 3 types now we discuss, but mainly there are 2 types of neurons. Okay, one is your sensory. That means one that brings, right? One that brings information from the sense organs to the CNS. That means this Praveen has touched a hot object, sense organs to the CNS, brain, right? That information is taken care by whom? By your sensory neuron. Sensory neuron. Okay. Now, Praveen has touched, but Praveen does not know what to do. That means CNS will send message to the effector, right? So, from the CNS, the information will be taken to the effector. Who will do that? Motor neuron. Motor neuron. Okay. Other than these two people, whom do we have? We have something called as association neuron. Association neuron. This association neuron is not completely sensory, not completely motor. Oh, it's not completely motor, not completely sensory. So, but still they do the most important functions. What are the most important functions, students? Questions have come from this topic. That is the reason why I am stressing so much. It helps for memory, communication, and intersensory association. Intersensory association. Clear? Easy? Full. That side I have written. Okay, only about neuron I have written on this side. Okay? Yes? White matter, grey matter, no doubt, right? White matter, grey matter, no doubt. Okay? So, students, if we just like one second. Okay. Students, if we just draw your brain like this. Oh, one second. Okay, brain like this. Just if we draw like this. Okay, your cerebral hemispheres, if we draw like this, this becomes your longitudinal fissure. Okay, longitudinal fissure. Now, both of this, okay, imagine left and right is there. It will be combined with corpus callosum. Okay, that is not what I am going to try to show you. After corpus callosum, they are going to be thalamus. Okay, then there is going to be hypothalamus. Okay, this is what I want to show you. This is the blue dots are hypothalamus. This looks like a smiley, no? This one is your thalamus. Okay, now we will understand diencephalon. This is what? Thalamus. This is hypothalamus. Students, don't you think these notes are very important? Will you download the notes or will you not? Should I send the notes or not? Tell me, do you think you will get better notes? Yes, no? Yeah, I always laugh at my own diagrams. Okay. I will draw, but it takes a lot of time. Today's class, full time I will be drawing only otherwise. That's why I don't concentrate more on drawing. So when I used to draw for my MSc also, I used to always take a lot of time, like one full night I'll be sitting and drawing one brain, one heart. So that much time I used to take. So now I have reduced my time, right? Okay, diencephalon we told. So today, students, we are going to study thalamus. Thalamus. Yes. Understood the location of thalamus? Understood? Corpus callosum, below that you have thalamus, below that you have hypothalamus. Okay? Yes, you should download. Okay? Don't be like, I can't download, I can't put my phone number. Why is ma'am asking me to write so much? Okay, so download the notes, keep the notes with you. Even if you have written your own notes, well and good. But see, here more colors are used, more images are added for your understanding. So this will be helpful. Okay? Yes, students, thalamus is a relay center. What do I mean by relay center? That means whatever impulse is going from the sensory to the CNS goes to the thalamus. From CNS to the effector goes to the thalamus. That means double way, right? Whoever is going through this way and whoever is coming through this way has to cross the traffic police here. That is how thalamus. So it is called as the relay center. Clear? Okay. Hypothalamus is actually, you will study more about hypothalamus in your endocrine. Okay, that means your chemical coordination. Students, we are going to finish human physiology one by one, okay, in this channel, and we will also do something for the board students, right? We have to help all of them, okay? So, students, thalamus location. Hey, that's nice, yeah, GP. I never thought about it. GP, Gopika. GP, ma'am, that's nice, no? GP. People shouldn't say ma'am has BP then later. Okay, GP and BP has too much connection. No, later their ma'am will tell that GP ma'am always has BP. Right. So students, hypothalamus is below thalamus. Okay, and it is actually a link between your nervous system and endocrine system. That's why I told you, you will study about hypothalamus more in your endocrine. Okay, yes or no? Evolution. Yeah, I'll take. Yeah, I know it's a boring chapter, but we will make it interesting, right? Yes. Now we will come to Sharat's question. Sharath was very concerned about hypothalamus, right? Hippocampus, hypothalamus and your nuclei and all that. You know, what is, ma'am? Emotions, right? Emotions. So we will learn about emotions. Okay, 1 second. So before that, we'll just finish this. So students, hypothalamus contains a number of centers which control body temperature. That means it helps in osmoregulation. Here, let's add here only. So it helps in, it has many centers, okay, which helps in osmoregulation, homeostasis. Remember homeostasis also we discussed. Homeostasis, right? All this hypothalamus will help. We will study more there also. Students, okay, listen here. Urge for eating, drinking, all this is because of your hypothalamus. That is the reason. Now I'm feeling thirsty, I'll go drink water, right? All that is because of my hypothalamus helping me out. Also, it contains several groups of neurosecretory cells which secrete hormones, okay, which secrete hormones called as hypothalamic hormones, okay. NCERT lines, very, very important, okay. Now, the inner parts of the cerebral hemispheres and the groups of associated deep structures, like, what is this? Amygdala. This one, your amygdala, hippocampus, and one more, nuclei. All these are helping for your emotional reactions, okay. I'll tell you what each of them are helping for. So, students, this first person is helping for fear. When you have any, like, you're scared of something or any kind of aggression, it helps you with aggression. Why do you get very angry at someone when they ask you to do something? You get angry at your parents or your teachers when we ask you to download the notes, right? That kind of aggression and fear will be regulated, okay, regulated. And then your hippocampus, again I don't have place. Okay, hippocampus helps with your emotions like your fear, sadness, happy, all that. Genetic you want after this, okay. And also mainly hippocampus helps with my students short-term memory loss, okay, short-term and long-term memory. My students are facing that, yes or no? Right, today you will learn in this class. Again next week you will ask, ma'am, do neural control and coordination, okay, short term and long term memory. Today I'll tell you download the notes. Tomorrow, two days later, you will ask, ma'am, where is the notes? So short term and long term memory mainly focused by your hippocampus, okay, hippocampus. Then students, even with your sexual behavior, that is also controlled by these people. So along with the hypothalamus, it is involved with regulation of your sexual behavior. That means any kind of attraction you feel to someone, any kind of, you know, emotions and feelings that you feel at every kind of your age, right? Especially when you're a teenager, you will have more emotions, you will have more that tendency of getting attracted to some person. All of that is controlled by your hippocampus and with the help of your emotional side of your brain, okay, clear? Yes, long-term memory, good. If you have long-term memory, I'm happy. So excitement, pleasure, rage and motivation, everything is controlled by these people. But this, I feel, this is one thing my students lack, motivation. Motivation is what my students lack. No motivation only. Every day come for live class means no ma'am, no motivation today. But if I ask you people to watch a movie, yes, full motivation. So this part of my students' brain is having a glitch. Yes or no? Yes or no? Yeah, exactly. They will see the syllabus video and ask what is deleted. Okay, they will ask the same teacher, are you teaching zoology? Even if I'm talking about a dog in front of me, like I'm keeping a real dog and teaching, like, students, this is the head, this is the, ma'am, are you teaching zoology? Is this an animal? No, ma'am, are you holding a plant? That's the kind of students we have sometimes. So this motivation part of the brain is fully gone. Okay. Yes. So this is the location of all of this. Okay, location of all of this. Okay, you can see here there are so many portions which are not readed for you, but hippocampus is there. Okay, thalamus is there, hippocampus is there, mammalian body, that is not written. Okay, amygdala is there. Okay. Yes, that's it. Okay. So I don't know why do they keep names of as if it is some Hollywood singer, right? Fear, aggression, okay? Not only that, even with your sexual behavior, motivation, okay? Yes, all of you are with me? I'm just writing it for your notes. They'll be like, ma'am put picture and she did not explain. So I'm writing it for your notes, okay? Long-term memory, short-term memory, excitement, right? All this you feel, no? Excitement. If you crack NEET, your excitement, your reward, feeling of reward, right? That and all will come, okay? Oh, don't remind me organism and environment, students. That is one place that I've lost my patience, right? I have been like, how many times do I tell? Now everyone are calm. Now every single teacher is teaching on YouTube. So everyone are like, oh, now we have to understand that this is part of the portion. Because I took first. In English itself, I took first. They kept asking me after every 2 minutes. Why are you teaching deleted portion, ma'am? Why are you teaching deleted portion, ma'am? Why would I come on a Christmas and teach deleted portion, right? So students, this I have added again only for your understanding, to understand each lobes. What are they doing? Okay, each lobes, what are they doing? Is it helping with memory? Is it helping with emotions? What each part of the lobe is doing, just for your understanding. But you do not have to know what to tell, like function of all of the lobes. But in case anything comes, for your understanding, okay? Yes, students, here midbrain. Okay, first we will draw one diagram. 2 eyes, 2 small eyes, nostrils, right? Nostrils. Yes, so your midbrain has 2 parts, correct? Midbrain has 2 parts. What are the 2 parts? Corpora, quadrigemina, and we have uncle, right? Cerebral, peduncle. Okay, please don't write uncle. Cerebral, peduncle. Okay, so let's see the structure. So students, here quadra or quadri means what? 4, right? 4. Okay. Quadra or quadri means 4. Yes or no? Right? That is what we are going to understand here. Students, this is called as okay. So your quadra means 4. So I have drawn 2 bigger circles. So let's just mention the superior and inferior. Then I'll tell you the name. Okay. Superior, inferior. Okay. Simple. Bigger one superior, smaller one inferior. Right. So upper one superior, smaller one inferior. Is this clear? Is this clear? Yes or no? Now this superior, it's called as colliculi. Superior colliculi and inferior colliculi. Okay, these constitute your corpora quadrigemina. Okay, the superior colliculi and inferior colliculi. Okay, now other than this, superior is going to help for visual reception. That means I am seeing the smoke. That means I know something is burning. Right, I am seeing that, I don't know what I am seeing. Okay, I am seeing that there is a lot of light here. That means I can see, oh, there is enough light here. I am seeing that from the tea the vapors are coming. That means it's hot, right? That kind of visual reception, right? Visual receptors. Now students, if visual receptors have to be there, that means optic nerve has to be there, right? Optic nerve has to be there. So this optic nerves will crisscross like this, okay? This is called as optic chiasma. Optic chiasma. Okay, this is going to help the superior colliculi for visual reception. Okay, visual receptors. Now inferior colliculi is going to help for auditing, right? Auditing. Auditory reception. That means I know that someone is entering. With the voice, I turn my head, right? That is controlled by my midbrain. Same way I can see that someone is coming in, so I look that side, right? So, or I'm writing like this, someone is coming from the back side, I turn like this, that's because of the midbrain. Okay, same way, someone opens the door, I look that side, again because of the midbrain. Clear or not? Visual reception and your auditory. Auditory means to your hearing. Okay, this is your midbrain. Okay, now let's understand your peduncle. Okay, what is this peduncle going to do? So students, your cerebral peduncle actually helps in connecting. Okay, connecting. Cerebral peduncle is a part of the midbrain, no one has that doubt. So cerebral peduncle will help. This is your FB, Facebook, okay, MB, that is your how much data you have, and HB. I don't know what is HB. Okay, so students, your peduncle, okay, is going to help the connection between your forebrain and hindbrain. Okay, it is going to help with the connection of your forebrain and hindbrain. Okay, that is what your cerebral peduncle is going to do. Clear? Forebrain, not FB, forebrain, midbrain, not MB, HB. Okay, forebrain, midbrain and hindbrain. So your forebrain will be connected. So students, you know this formula, no? AB is equal to BC, like that, AB, BC, like that. So this will indirectly or connect with your hindbrain. That is done. Okay, that is done by your cerebral peduncle. Right? Uncle means connection, makes connection. Uncle, aunt, what do? They bring the connection to the family. Right? Same way. Yes, students, only this much is mentioned in your textbook. The midbrain is located between where? The thalamus and the hypothalamus of the forebrain and the pons of the hindbrain. A canal called the cerebral aqueduct passes through the midbrain. The dorsal portion of the midbrain consists of mainly 4 round swellings. That is what I drew. Okay, called as the corpora quadrigemina. Okay, corpora quadrigemina. Clear? That 4 swellings, superior and inferior. Yes. Last part, hindbrain. All of you tired? Tired? Okay. Not tired, no? Okay, let me draw one thing by then. Don't be tired. We have some surprise. You will see all of our teachers together. After launch, you people didn't see us together, no? Okay. So students, hindbrain consists of what and all? Let's write here. Hindbrain consists of 3 people. Who are the 3 people? Cerebellum, pons and medulla. And this medulla will go to the spinal cord. Correct? This also has the grey matter, white matter, everything is there. But in medulla, right? In the medulla, the white matter and grey matter is ulta. Ulta means what? Who was inside? Who was inside? Look here, look into the slide. No, this slide. Look here, students. Outer we had grey matter. So outer is white matter in the case of your medulla oblongata. Okay, medulla oblongata. What is outside? White matter. Okay, this is the specification that you see in your spinal cord also, right? Outside you will have white matter and inside you will have grey matter. That is why you have seen that head-shaped structure, no, in the section of your spinal cord. Okay, inside you will have grey matter. So from the medulla, spinal cord is formed. So even medulla will have the same thing. Okay, what I have drawn here is ginger. It's not ginger, it's your cerebral hemispheres. Okay, cerebellum. Okay, right. So sorry students, cerebellum. Okay, 2 parts. It's divided into 2 parts. Okay, this portion, no, this portion which have you seen ginger also looks like this. That mid portion is called as vermis. Okay, these are your hemispheres again. Cerebellary hemispheres. Okay, tongue twister is happening in my case. Okay, yes. Please explain my brain diagram. You mean my brain diagram? You want me to remove it out and show and teach? Or is it okay like this only? Yes? Yeah, it looks like ginger, no? But it's not. Okay, it's not ginger. Students, gray matter, white matter, clear? Gray matter, this is important, okay, this is important. Spinal cord, gray matter, white matter is important. Now, cerebellum is the second largest part. Cerebellum helps for, have you seen people who drink alcohol, they go zigzag. Have you seen they go zigzag? Why do they go zigzag? Because the cerebellum lost the balance. Cerebellum helps for balance. That is the reason why we are able to walk. Even if I draw a line, you will be able to walk. Same way, have you seen people, have you seen people doing very, learning things? That means like cycling. Okay, cycling is an example. When you learnt it the first time, you were thinking about should I keep the pedal up, down, all that you were thinking. But as time passed, you do not think so much, right? Automatically, if you see a person in front of you, you will horn, right? Same way, if you have to take a U-turn, you will turn. All those things at the starting, that starting learning part came from your cerebellum. Okay, learning part came from your cerebellum. Is this clear? Same way your cerebellum works with balance. Why I told that is, students, have you seen these people in circus walking on a rope? Have you seen how carefully they balance their hands and walk? That is because of, again, cerebellum. So cerebellum helps for balance. Okay, balance. That is why a person who drinks alcohol cannot walk straight. They walk zigzag. Okay, and they are also helping with finer, like accuracy. Like that accuracy comes because of cerebellum. Okay, accuracy. Students, not only that, the learning part. That means if you have to learn some skill, right? If you have to learn some skill, that starting learning part of that is done by your cerebellum. Okay. When we spin, so when we spin also, what happens? It is difficult for the impulse to be transported, right? Because you're continuously holding like this with your friend and you're turning. After some time, you lose balance, correct? That is also because of that movement. Okay, that is the movement. Okay, clear? Yes or no? So all the involuntary activities also will be taken care by your cerebellum. Okay, involuntary activities, right? Cerebellum. Okay, your cerebrum will help with what? Thinking, memory, intelligence, all that your cerebrum will help, right? Cerebrum will help. Medulla oblongata will help have all the centers, cardiac center, vomiting center, thirst center, all that will be taken care by breathing, all that medulla oblongata will take care. Okay, so yes, I think that's it. That's it, students. Hindbrain, medulla oblongata, I'll teach you in the next slide. Okay. Yes, there is also, for your understanding, the most complicated or the most complex neuron called the Purkinje neuron is present in the cerebellum. Okay, Purkinje neuron is present is because of the scientist who came up with it, Purkinje. So, Purkinje neuron is present in the cerebellum and it is one of the most complex, right? Most complex and most efficient neuron. So it is the most complex neuron. Okay, with me? Yes, all of you are with me? Easy? Is there anything that you're finding it difficult? Students, this structure, no, if I take a half of this, right, if I take half of this, it somewhat looks like your, have you seen the Christmas tree? Not the pine tree, not the pine tree that you see, but have you seen the ones with like proper green one? You would have seen it in Orient Mall, you would have seen it in so many malls, they will keep them. It's called arbor vitae. Arbor vitae. It is called, this is called as arbor vitae if you take a longitudinal section, because it looks like a leaf of the arbor vitae tree. Okay, it will look like that, right? It will have a ending. So if you see that tree, no, it will look like this. It will look like your Christmas tree, like this it will look. Okay, like this, arbor vitae. So this cross section also will look like this. It will have a tip at the end and it will have lines in the middle. Okay, that is why it is called tree, and it is also called a tree because it is also known as tree of life, right? Arbor vitae, tree of life. So that's why this is known. So students, if you see here also, this will also look like Christmas tree, right? Arbor vitae. Okay, tree of life. Got it? Any doubts? No, right? Simple. Okay. Oh, 1 second. Yes, students, medulla oblongata also last topic. Okay, so Pons helps with respiratory system. Okay, pons help with respiratory system. Everything to do with respiration and whatever systems are there, that will be done by your pons. Okay, medulla oblongata. Have any of you heard this? Here is your medulla oblongata, right? Here. This portion is your medulla oblongata. Have you heard that when someone punch or when someone hit you in the medulla oblongata, you might end up dying, right? Have you heard that? That is because your medulla oblongata has, the pons helps with all the respiration, right? So when you hit here, even your pons gets affected. And medulla oblongata has your cardiac systems, cardiac centers. So that is the reason. Everything to do with your breathing, cardiac, your heart, all that is controlled by your medulla. That is why when someone punched there or hit there, right, punch there or hit there, there are chances that you end up choking and die. Okay? Okay, got it? Yes? Yes or no? Any doubts so far? Any doubts so far? I'm checking the likes. All of you, did you all like the session? I'm sure most of you liked it. Most of you liked it. Yes? Clear? Very clear? Yes. Okay. Why is Purkinje neuron most efficient and complex? So this Purkinje neuron is most efficient and complex is because of, like how I told you, they are because of their structure. That means they are very specific. They are very cerebellum specific, right? They are very specific and they are very complex because of the way their structure is. That means their structural diagram. Structure, that means they have more branching, and those branches are efficient. That is the reason why they are more complex and why they are more efficient. Is that clear? Who asked me the question? Sharath? Is that clear, Sharath? Yes? Okay, they are branching, they are amount, massive, they are very massive in amount. All this makes them very complex and special about them. What is more special about them is because of this complexity or their branching and all of that. Okay, yes, students, so medulla oblongata, I told you it has a lot of centers by itself, right? It has a lot of centers. So those centers, what and all centers can they be? Cardiac center, okay, which will help cardiac, one second. Cardiac centers. Okay, they have centers which will cause the vomiting. Okay, then they have hunger, like hunger, thirst, all that will be controlled. Okay, right, vomiting, tendency to vomit. Okay, that is done. Again, respiratory centers. Okay, here also centers. Okay, then for your this one, student, swallowing, you swallow your food, for swallowing, even your medicines, when you want to swallow, all this medulla oblongata helps. That is the main reason why they say if anyone hits you here, it's not okay. Have you seen this? Some people have a tendency to hit, right? When they tell hi and all, they come and hit on the back. But if in case they hit very strong near the neck region, it can cause a problem. Okay, it can cause a problem. Clear? Is this clear? Any doubts so far? No doubts? Okay, let's also see the NCERT ones and then we'll be done. Okay, in case we miss something, we will just see the NCERT. Oh, it's dancing. Okay, yes. Students, look here, the midbrain consists of the pons and the cerebellum and the medulla. Okay, also called as the medulla oblongata. Okay, pons consist of fiber tracts that interconnect different regions of the brain. Okay, it connects different regions of the brain. Cerebellum has a very convoluted surface in order to provide additional space. That is what you see your brain like this. No, have you seen like this? Like it will look like this. So many of them, right? That is because of your cerebellum, to give additional space for many more neurons to come. So more the neurons, they say you are more intelligent, right? Okay, and the medulla of the brain is connected to the spinal cord. That is the reason why your medulla has outside white matter, inside grey matter. That means your grey matter, your cyton and your axon endings are inside. Your axon alone with the myelin is outside. Okay, the medulla contains centers which control respiration, cardiovascular reflexes and your gastric secretions. Three major regions make up the brainstem: midbrain, pons and medulla oblongata. Brainstem forms the connection between the brain and the spinal cord. Okay, this I already mentioned you in the starting itself. Any doubts? No doubts? Clear? Right, students? Clear? Thank you so much. Start new life. You have started your new life. Okay? Yes? Students, done or done? Can we have rapid fire? Rapid fire. Okay, rapid fire. Ready? Let's put fire emoji in the chat box for rapid fire. Then we are done. Then we are done. Right? Easy chapter. Did I make it easy? Okay, first question. First question. Okay, rapid fire. Let's see. Very good. Look at the chat box. It's lighted up. Right? Okay. I'm just going to draw something. You have to tell me this. Students, tell me which type of a neuron is this? Which type of a neuron is this? First question. Okay? Which type of a neuron is this? Wow, very good. Bipolar. My God, everyone answered. Good job, good job. Very good. Okay, students, second question. Second question. Tell me which part of the brain is the connecting link between nervous system and endocrine gland? Oh, Subhashni, very smart. Subhashni already answered. Other people are still typing bipolar. Very good, students, very good. So many of you answered bipolar. Okay. Yes. Hypothalamus. Right? Hypothalamus. Okay. Now, third question. Ready for third question? Okay. Okay. Look at this. Look at this diagrams. All diagram based questions today. Okay. Students, I want you to label this, this and this. 1, 2, 3. 3 questions at one shot. 3 questions at one shot. Come on. Go for it. Which is the first, second and the third part? 1, 2, 3. Let's see. Gyri. Okay, first part is the gyri. Yes, very correct. Amazing. Second part. What is the second part? Fissure. Very good. Third part. Sulci. No, Lakshmita ulta. Second part is fissure. Third part is sulci. Right? Okay, fourth question. During repolarization, which channels open? During repolarization, which channels open? Which gated channels open? Yes, correct. Which gated channels open? During repolarization, which gated channels open? Very good, Rupa. Very good. Very good. Let's see more answers. Let's see more answers. Yes, very good. Potassium gated channels. Potassium gated channels. Okay, fifth question and the last question. Fifth question and the last question. If the meninges are read from inside to outside, what is the word? Inside to outside, the meninges. Inside to outside, if I move, how will the meninges order be? What will be the order of meninges? Very good, very good. Potassium gated channels. Good job. Good job. Students, inside to outside, if I am reading the meninges, what is the order? Good job, good job. Amazing, amazing. Wow, just looking like a wow. Right, chat box just looking like a wow. Pad, very good. Pad. P stands for pi matter, A stands for arachnoid, D stands for dura matter. Yes? Good job. So, all of you answered all the questions right? Bipolar. Yes. Rapid fire. Very rapid it was. Very good. Very good. Okay. So, we are done. No doubts, right? Is there any doubts? No doubts? No doubts. Great. Okay. So, you can fill the chat box with red hearts. You can fill the chat box with red heart, or you can fill the chat box with brain. Brain. Okay, your choice. Brain or heart. Yes, correct, Ananya, correct. Brain diagram you want to explain? Which diagram? 1 minute, Shena. This one? Shena, this is only the lobes. So students, you have frontal lobe. So here, this you will study in locomotion. Frontal lobe, no, this way, no. Frontal lobe, okay, frontal lobe is here. Then you have parietal, then you have occipital, then you have temporal, right? So the lobes, each lobes help for one part. So brain diagram as such you don't have, you only have the parts. So here if you see, you can see frontal lobe, corpus callosum, okay, hypothalamus, which is there, medulla oblongata is there, pons is there, spinal cord is there, cerebellum is there, thalamus is there. Okay, that's all. Wow, beautiful chat box. Look at the chat box, how beautiful it looks. It's so pink and beautiful, right? Yes. So students, do not fail to download these notes. It has everything that you need. And where will you download the notes? You will download it on our Telegram channel, Vedantu NEET English Telegram channel. Weekly schedule will be there. Mock test will be there. That means your proper NEET mock test, like how you will write for your 720 marks, right? You will write for 720 marks. Exactly like that. Every Sunday mock test will happen. Sometimes it will be from 12 to 3. Sometimes even it will go till 3 to 6. Okay, so weekly schedules you will get on our Telegram channel. Daily quizzes. Yesterday and today morning also quizzes were there for this chapter, neural control and coordination. Every quizzes is from your previous year NEET questions. Okay, so every time you answer one question, you are getting 4 marks in case that question repeats in your NEET. Okay, weekly live sessions. Okay, Shreyas sir will come weekly and he will give you motivation that is required for you, right? And then you have daily session PDF. That means this PDF you will not get it anywhere else, not even on my Telegram. You will only get it on Vedantu NEET English Telegram. Okay, you have to log in with your phone number and OTP, and you can itself write on the notes. Wherever I have left blank spaces, you can write. You can mark on the diagrams and learn. Okay. Weekly mock test will be there. That means we are starting, or we will introduce you to weekly mock test, where every week you will have test. That is your Sunday mock test, right? We introduced you last time. That mock test happens. Super 6 series, that means every day live classes to complete your syllabus as quick as possible. These things you will only get on our Telegram. Okay, this is how it will look like. This is how your update is going to come. This is how quizzes will be there, announcements will be there, and your notes will be there. Okay, and students, to start the quiz, you have to put your email ID, then you can click on start test and end test. That means you will get 3 hours exactly like your exam, 3 hours 15 minutes. You can read the paper and you can start attempting. After attempting, mock test results will be published on the Telegram channel itself. You can see where your scores are. Last time amazing scores had come. Lot of students like Sanvi were there. So many of them were there who scored really good marks, like 590 up to 590 they had scored. So do attend the test. And then students, Telegram quizzes also will be there, where you have to click on start, attend the quiz, and your answers will come correct. Okay, if you're I am ready, attempt the quiz, score and rank will be shown among all the people who have attended. Okay. No, immunosuppressants, I don't think it should be. Students, do not believe Google every single time. Okay, there are a lot of mistakes. People enter by themselves sometimes answers on the, what to tell, the Google. So do not believe Google every time. Okay, so sometimes here and there they cook up their own story, right? That's why I told you last, like when you check any symptoms of your diseases, they'll end up telling you have cancer. Correct? Okay, so students, no, Shrenas, no, do not be sorry. You just asked me a doubt, but it's not there in your syllabus. Like frontal lobe, you just need to know the lobe's name. You don't have to know the correct position, because locomotion and movement you'll study in detail. Okay, students, and also 7 years your DPP quiz awaits. That means it's there in the description. If you go to the description now, you will see 3 links. Okay, 3 links you will see. Click on that links and you will be directly taken to, see students, look here, this is our live class happening. Right, this is my class. So if you click on more and if you go here, you can see try solving 7 years PYQ quiz. Students, it's very important to solve your PYQs. All of you know that, I hope. See, here you enter your phone number, click on next, or you enter your email number, click on next, you will get an OTP. After the OTP, you will be directly led to the quiz. Okay, and if you want to join our Telegram channel, you have to click on this Vedantu NEET English. Okay, directly you will be taken to our view in Telegram. I have to download now, but otherwise you can just join the Telegram. Clear? Yes, no doubts, right? Everything is here, all the links are here. See, it's very precise, so you don't have to break your head. You can just go click on it and you're done. Okay? Yes? Yes. Clear? Yes, so thank you so much, students. We are done, right? Chats are moving. Let the chats keep moving, right? Let the chats keep moving and moving. Lots of love should be there for your this session. Yes, great. Okay, thank you so much, students. I hope neural control and coordination is super easy for you from now on. No more you will have to be like, ma'am, I can't understand. There's so much about brain. There's so much about that, this. And do not fail to download the notes. I've added so many images for you people, just for you people. I do not need images, right? I do not need images. I just put the images for you people. Okay, so you people can solve it and use it. Okay. Yes, so many reflections are coming. So, okay, students, thank you so much for joining. See you all in the next class. Do not forget to comment which was the most favorite part of this session. Put the time limit. I will also see your timestamp. Let me see which was the most important or the most lovely part of this class. Thank you so much. Do not fail to like, comment and share. See you all in the next class. No. See you all in the surprise session. We will see you soon.